everyone. Welcome to the second virtual pre-conference of our Microphysiological Systems World Summit series. So the first MPS World Summit will be a hybrid event in New Orleans from May 30th till June 3rd of 2022. We very much hope to meet you there again, and a big thank you to all sponsors for their financial support. I'm Tamara Zitek from Doctors Against Animal Experiments, representing the Eurogroup for Animals, and I will moderate the first part of this event. As current chair of our virtual event committee, I want to thank all committee members for their valuable help in organizing today's pre-event. Thanks also to all speakers and panelists and to the Scientific Advisory Committee for their support and for selecting today's speakers from the abstracts. I would like to remind all speakers to stay in time, please. Amongst our speakers, we have 4 young investigator oral presenters who are the finalists from 22 applicants and will be competing for the Young Investigator Award. The voting for the award will take place after the second short talk session, and the winner will be determined by the audience. So we would like to encourage all of you to please vote for your favorite. The winner will finally be announced after the panel discussion. Please use the Q&A feature to submit your questions to the speakers. If we won't have time to answer questions after the talk, they will be answered in the Q&A box. One quick note that all of you received an email recently where you find information on how to access the posters. The link to the posters and the password will additionally be provided in the chat box. Please address your questions to the poster presenters directly via their email. Okay, so now I'm passing the mic to Thomas Hartung from the Center for Alternatives to Animal Testing. Thomas, the floor is yours.
Thanks a lot, Tamara. Thomas Hartung from Baltimore speaking. It's my pleasure to welcome you all. This is the next step towards what we call the first World Summit on Microphysiological Systems. Okay, so this is the second step we're actually taking, the second pre-event. As you just heard from Tamara, we are thrilled to welcome you in New Orleans, hopefully in person, but also possibly in a hybrid version. And I'm very happy that with Don Ingber, one of the co-hosts of the New Orleans event, is going to give the keynote like Susie Fitzpatrick did for the event in the last pre-recording. This all started with a workshop in Berlin where we all came together, and over a beer with Dan Tagle from NCATS, the idea was born that we need international conferences and possibly an international society. I'm thrilled that in September this led to a paper in Science which is summarizing the ideas of this workshop, and I invite you to read this to understand what the concept and the opportunities of these technologies actually are. We have brought around 45 organizations behind us who are supporting this event. You can see them listed here and in our regular communications. I have no time to mention them individually. But there's also quite a number of individuals who have committed their time in the various committees in the Scientific Advisory Committee, the Fundraising Committee, and our Executive Committee. You have heard we have sponsors who help us to make this happen. The NIH with NCATS has done a major contribution to make this all happen. Please consider supporting our series of events, because we are still far away from our goal to really finance fully the event in New Orleans. So your logo will find a place quickly on the list of sponsors if you only contact us. The first pre-conference is history, but you can still follow it on our YouTube channel, so you can get information about this. It was devoted to regulatory acceptance. Today, you will hear in our second conference more about the engineering side of microphysiological systems, and you have the program all with you. New Orleans has already a pre-program which is available. You see the topics on the right, which we will address. Please consider submitting abstracts. We are going to open this instantly. Tomorrow we will launch our new website, which gives more information on this. This is only the first of a series of events. We are planning to bring the conference a year later to Berlin, and already a committee is working on the program, and then a year later to the west coast of the US. So please see this as something where you can join a family, where you can become part of these Microphysiological System World Summit series. And with this, I wish you all an exciting day. I hope that we learn a lot that the community for microphysiological systems is forming through this series of events. Thank you very much.
Thank you, Thomas. So now we have our keynote speaker, Donald Ingber. We are very honored to have you here, Donald, as a keynote speaker, and we are very much looking forward to your presentation.
Thank you. I'm going to share my screen and hopefully you can see this.
You're good.
Great. Thank you, Tamara. And also, I want to thank Thomas on behalf of all of us for all the hard work he's done in building this MPS Summit series that's really bringing the field to the next level. So thank you, Thomas. I'm really happy to have a chance to share with you what we've been doing over the past many years in terms of exploring human organ chips as tools for therapeutic discovery and now clinical mimicry as well at the Wyss Institute in Boston. When we first started to pursue this path, one of the major drivers was the recognition that the drug development model is basically broken in that it costs a huge amount of money to bring drugs to patients at this point, and this has many reasons, but a major feature is that animal studies are costly. There are ethical issues, but the real problem is that 70 to 95% of the time, they actually are wrong when it comes to predicting clinical responses in clinical trials, and this leads to major failures. But over the past 10 years, we've come to realize there are many other drivers behind this field. For example, right now, 40% of drugs in the development pipeline are biologics. These are therapeutic monoclonal antibodies, now CRISPR and RNA-based therapeutics. And many of these are so specific for human targets that there really is no animal model. Non-human primates are still used. They're the closest we have. But as you see at the image at the left from New York Times earlier this year, there are now shortages of these because of vaccine testing. Furthermore, even non-human primates often don't predict what happens in humans. So another real need for preclinical human models. Another driver that's emerged is the big leap in microbiome research, where we now know that microbiome plays a central role in both health and disease. And there just are no good test beds for human microbiome-based therapeutics, because not only is every species microbiome different, but even every individual. And finally, particularly with the COVID-19 pandemic, it's becoming more and more clear that we have what they call a One Health challenge, which is that many of the vectors of diseases emerge in lower animals, such as bats and SARS-CoV-2. And there's no way that you're going to be able to develop drugs for these animals to prevent the emergence of disease by testing every animal in a laboratory. However, there's this possibility of using organ on chips to develop species-specific models. So we first coined the term organs on chips because we set out to engineer microchips containing living human cells that reconstitute organ level, not cellular tissue, but organ level functions to accelerate drug development and replace animal testing, but also to advance personalized medicine. And we called them organs on chips because we used computer microchip manufacturing methods to enable this. And George Whitesides and I had started to bring these methods into cell biology in the late, around 1990, so over 20 years ago. Now, our first breakthrough, I think many of you know, was a paper in Science. Dan Hutt, when he was a postdoc with me, was the lead on this, developing a human breathing lung on a chip, was a model of the alveolus or air sac. And the next video shows you how our chips work. So they're the size of a computer memory stick, optically clear, made out of a flexible silicon rubber. In the center, there are 3 hollow channels, each less than a millimeter wide. Oops, sorry here. Let me go back. If you cut it in cross section, you'll see that there are 3 channels, each less than a millimeter wide. The middle channel is cut into top and bottom by another thin flexible membrane that has pores that allow cellular motility. It's coated with extracellular matrix, and to make the lung alveolus, we put human lung cells up top, human lung capillary cells on the bottom, and we thereby recreated the alveolar capillary interface. And then the trick for the lung is that we had cyclic suction through these side chambers. Everything was flexible, and that replicated the mechanical microenvironment in terms of breathing, the degree of strain and the frequency. We then could create an air-liquid interface. We could flow medium through the bottom channel, and now we have endothelial cells on all four sides, so we could even flow blood for short times. Now, if this were to work, it should model organ-level physiology. If you have a bacterial infection, you have the epithelial tissue will give out cytokines, produce cytokines that then activate the endothelium to express adhesion receptors, and white blood cells that normally just flowed by now stick, roll, diapedese, migrate across and engulf the bacterium. So now I'll show you imaging in the device. The immune cells are white, fluorescently labeled. You can't see the endothelium, and the epithelium is behind the screen. To begin with, quiescent endothelium, they just flow by. Now you add bacteria on the opposite side. Now you get this tissue organ level signaling response. For example, ICAM is expressed. The white cells are pulled out under flow, which is very important for initial adhesion. And now you can do any imaging in these devices. You could do in vitro or in vivo. So here's one immune cell about right there. It's going to find a space between two unlabeled endothelium. It then goes out of focus as it migrates through the matrix-filled pore, pentagonal pore. And then here's phase contrast on the other side of the cell emerging from the epithelium. And now I'm going to show you the white blood cells in red, because the bacteria are in GFP green, and you watch them in being engulfed. So you just watch the entire human inflammatory response in this little rubber chip. Now, I don't have time to go through it, but we have multiple publications where we've been able to use that specific lung alveolus chip to model diseases like pulmonary edema. We're now doing pulmonary fibrosis, drug toxicity, drug efficacy. We've discovered new therapeutic targets. There's drugs in clinical trials that came out of this. We've even developed gene therapies in these chips. And we've integrated immune cells, as I showed you. We've integrated stromal fibroblasts. This is synthetic biology at the cell, tissue, and organ level. You could add cells and more complexity if you can't mimic a response. But if you do mimic it, you've just learned something, which that it may be simpler than you thought. This is one in numerous awards. I won't go into those. But we started to explore other organ chips, and we in the lung, we basically then moved and we built a lung airway chip, not an alveolus chip, and this we could show reconstitutes normal ciliary motion, directional motility, normal mucociliary transport rates, and we started to then make these chips with cells from patients with diseases, and we did this with chronic obstructive pulmonary disease, COPD, first. And what was interesting that even though this is an inflammatory disease, we're only taking the epithelium. There's no immune cells. We put them on a chip for 3 weeks at an air-liquid interface. They differentiated, as I just showed you. And impressively, they retain the COPD phenotype, meaning there must be epigenetic signatures here that are stable. They have decreased TOL-R3 and 4 receptors. Now, what brings a COPD patient to the emergency room is exacerbations by bacterial infection, viral infection, or cigarette smoke. And in this early paper, we used mimics of viral infection, poly-IC, or bacterial infection, LPS endotoxin, and we could show that the COPD chips were in fact more sensitive. They were exacerbated, if you like, in terms of IL-8 with LPS endotoxin, or MCSF cytokine you could see with a viral infection, but the healthy did not show this. We then started to explore cigarette smoke. Candace Benham was a postdoc at the time, and with the engineering teams at the Wyss, built a cigarette smoking robot. This is a cigarette lighter from a car, a Gatling gun with real cigarettes. And this mimics the smoking parameters, the breathing pulses and pauses and intensities. And real smoke, not extract, real smoke goes all the way to the airspace of the chip. And just two interesting findings. The first was that we could confirm that cigarette smoke definitely exacerbates the COPD chip, but not the normal when measuring IL-8, a major inflammatory cytokine. It almost doubles in levels. And then we started to do transcriptomic analysis, and we have now done proteomic, glycomic, metabolomic, and transcriptomic on these chips. And here, the rate 3 lanes are 3 different healthy patient airway chips, the change in gene expression after exposure to cigarette smoke. The 9 at the left are from a publication, a clinical study of otherwise healthy patient who smoke cigarettes. And you can see that the top quarter versus bottom 3 quarters are pretty similar. We were very excited. The reviewers had problems because ours were too similar to each other, and there was greater variety in the patients. And then we realized that with organ chips, you could actually do this sort of clinical study better than a clinical study because you're doing matched comparative modeling in the same patients before and after a stimulus. Those changes are due to cigarette smoke exposure, whereas in these patient populations, some are due to cigarette smoke, but many are due to, work history, work home environment, genetic variations, et cetera. And so this actually is something we never expected in the beginning. We just published a paper last month in making chips with cells from patients with cystic fibrosis. And as you can see in the top, you in fact get greater ciliation of the surface. You have a higher beating frequency, not seen in static transsols, only seen with flow, increased mucus production, increased immune cell recruitment or baseline inflammatory state, increased cytokines, and increased overgrowth of Pseudomonas aeruginosa, which is part of the microbiome in all of our lungs, but overgrows in these patients. And so these definitely are fantastic tools for preclinical models that are human relevant. But what we realized over time is that these are more than tools for just looking at biochemical activities or mimicking tissue-specific functions. They really provide a window on molecular scale activities inside living cells within a relevant tissue and organ level context. And thus, we really believe that they can enable mechanistic insights and drug discovery. And so just some examples. 4 years ago, we were funded by both DARPA and NIH to leverage our airway chips to develop models of viral infection that could be available if there were ever a future respiratory virus pandemic. The focus was on influenza at that time. And so we actually showed that we have a beautiful model of influenza virus infection where you see at the bottom GFP virus infecting at the left, you disrupt junctions, you lose the ZO1, cilia are disrupted. We even see endothelial injury that has not really been studied much in the past. We could show differences in virulence in different viral strains, which is matched what you see in patients. We could quantify barrier function either using fluorescent dyes or inline sensors we've developed for transepithelial electrical resistance. But more importantly, as opposed to cells in a dish, you can analyze human host responses to infections and drug therapy. So at the top are showing a cytokine storm, if you like, IL-6, type E10, rantas, et cetera, in response to different viral strains, and the levels and degree of response scales with the level of virulence of these viruses in humans. We could also flow in immune cells, and not only do we see them migrate to the airway channel, even though they were flowed through the vasculature, we see them clear the cells that are infected with virus over time. And finally, we started to test drugs like oseltamivir, which is Tamiflu, the first-line drug in influenza, and we could beautifully see inhibition. We just published a paper 2 weeks ago where we showed that we could now use this to study evolution of virus variants that are resistant to therapy. And we did this by infecting one airway chip with influenza and treating with a drug at 90% efficacy. And then we basically take a droplet, wash it through the airspace, and pass it to another chip, just like when a human was coughing and there was human-to-human transmission. And we do that in the constant presence of drug that's 90% effective. With that, using amantadine, a clinically used antiviral, although not used that much anymore, you can see in 8 passages, you see emergence of variants that are completely resistant to therapy. Using Tamiflu, we did the same thing, but it took 25 passages. The interesting thing is that when we did viral genome sequencing, we identified all of the known mutations that have been published in patients who have become resistance to amantadine therapy, plus 3 new ones that no one's ever seen before. So you can now potentially use these to predict what's coming down the pike, and obviously could be very useful for COVID. Now, when COVID hit in January of 2020, I have to move my cat, we immediately pivoted. My Long C and Haiqing Bei, who are my postdocs, were virologists, and from China, were aware of this from social media. And within 1 day after the gene sequence being published in January, we engineered, or they engineered, our own SARS-CoV-2 spike protein expressing pseudoviruses. So because we only have a BSL-2 lab, they quickly started to work with the conventional cells that virologists use. We used HUH7 cells where they know viruses grow well. And they started to test about 30 drugs that had shown some efficacy in publications against related viruses like Ebola, MERS, and SARS. And as you can see in this slide, out of those 30 or so, there were 8 that in gray showed dose-dependent inhibition with existing drugs. The idea was, could we repurpose an existing drug quickly in the low micromolar range? In this group of 8 were, and this is January of 2020, chloroquine and hydrochloroquine. And so we then moved to the organ chips. And the reason we did that is that the cell lines people use, HUH7, Vero6, often don't have normal interferon response, and they don't express high levels of ACE2, the receptor for SARS-CoV-2, or TMPRSS2, the enzyme that's important for entry. We confirmed that there were high levels of expression of both in our differentiated human airway chips. And now we tested those same 8 drugs, but we flowed them, perfused them through the chips at a clinically relevant dose. We didn't just bathe them in drug like we usually do with static culture. And now hydroxychloroquine and chloroquine had no efficacy. The 3 at the right that failed in clinical trials had no efficacy. And we had 3 drugs that showed some level of efficacy, with amodiaquine, another anti-malarial related to chloroquine and hydroxychloroquine, was the most active. And so we pursued this with funding from DARPA in collaboration with Ben Tenover at Mount Sinai and Matt Freeman in University of Maryland. And long story short, we found that in a hamster model of CoV-2, that amodiaquine could prevent infection significantly when given 1 day before infection, where hydroxychloroquine was totally ineffective. And that has been, hydroxychloroquine effect has been confirmed by many labs. If you look at histology, you see a major suppression of infected cells. We had an over 95% inhibition of animal-to-animal transmission, and it worked even if you gave the drug 1 day after treatment. And based in part on our results in organ chips, this drug is now in clinical trials across 20 sites in Africa. And more recently, we extended this work to our human alveolus chip. This is Haiqing Bei's work. And we used H3N2 influenza, which affects the small airways, causes pneumonia, and is more virulent, higher morbidity and mortality in patients. And you can see in the top that you get very nice cytokine response. You also can see that that the host responds by putting out interferons. That is our way of fighting our own antivirals, if you like. But what was really interesting, and this could only be seen in our organ chips, is Haiqing explored with or without breathing motions. And he found that physiological breathing motions suppressed viral infection, prevented viral infection. So mechanical strain is breathing motions. And you can see decreased viral RNA levels, decreased titer, decreased cytokines. And what we found is that breathing motions stimulate type 1,3 interferon responses, as shown at the left. If you compare Transwell, and here in the blue, two weeks with physiological breathing, we have high induction of multiple interferon stimulated genes. And we do RNA-seq, we found the same, but we also found that the most highly expressed protein was a protein called S100A7 that was not induced in Transwells. And this is not only RNA-seq, but secreted protein. And we then overexpressed this protein in cells, and we found that it actually induces the interferon response in human lung alveolar cells. and this across multiple innate immune responses. And then we found an inhibitor, I should say that S100A7 binds to RAGE, which is a receptor for advanced glycated end products, is a major mediator of inflammation, and the alveolar cells in the lung have one of the highest levels of expression of it. Also endothelium express it. And we found when we used a drug that was in phase 3 clinical trials for Alzheimer's, but targets RAGE, we almost completely suppressed the inflammation cascade, the cytokine storm, if you like. And more recently, we also combined it with molnupiravir, which is the Merck drug you may have heard about that showed a very positive response in 30 to 50% of patients with COVID-19. And here you can see that this drug alone gives you some suppression of cytokines, actually minimal of some, but we synergize with it by using this RAGE inhibitor. And we are now working with Cantex Pharma, just about to submit an IND to begin to ask to initiate COVID-19 clinical trials with this drug as well. We've also discovered entirely new drugs using organ chips. So we used a CRISPR-based screen to explore RNAs that may mediate response to influenza infection. And it was a bit of serendipity here, but long story short, we were able to discover a novel class of immunostimulatory duplex RNAs that are short, about 20 base pairs, that have a unique sequence motif. And these turn out to be incredibly broad spectrum antivirals. So we now, these induce interferon beta, for example, many-fold in human airway epithelium and alveolar epithelium. When we tested against H1N1 and H3N2 in the airway chip and alveolus chip, we get potent suppression of viral load. But this has now been also been shown to be active against SARS-CoV-2, MERS, and NL63, which is the common cold virus. So incredibly broad protection by inducing interferon responses. Okay, so that was lung. We've made many other chips. This is a human intestine chip. We originally made it with cell lines, but now we take organoids from humans, we break them up, put them on the top channel, put endothelium on the bottom, and now we can do absorption, we can measure transport, and when we give peristaltic-like motions and flow, we get beautiful intestinal villi formation. We have published that transcriptomic analysis shows that these chips are closer to the human transcriptome than the organoids from which they are made from. We've now been leveraging these chips to get into microbiome research. First thing we did was a collaboration with Lacroix's group in Switzerland. We were exploring why different microbiomes from different species, whether they contribute to the species-specific tolerance to infection. For example, enterohemorrhagic E. coli in humans, a small number of bacteria will cause disease, and mice incredibly resistant, orders of magnitude more bugs and they don't notice it. In this case, we just grew up microbiome in bioreactors, took the metabolites, put them on the chips. We could confirm that if you have yellow are cell staining. So if you had human metabolites from the human microbiome, major injury when you give E. coli, mouse, minimal. And we used metabolomics in this study. And long story short, using metabolomics, we were able to find 4 specific chemicals produced by microbiome, metabolites in red arrows here, that are responsible for this effect. I'm just showing you that these are great discovery tools. More recently, we went much further, and we've now developed models where you could, for the first time, co-culture human cells with living complex gut microbiome, both anaerobes and aerobes. We did this by creating a hypoxia chamber so that we can get a hypoxia gradient across the tissues. So with high oxygen in the vascular lumen that you perfuse with, no oxygen at the apex, we actually use nitrogen gas. The human cells survive, but you can keep anaerobes and aerobes in the lumen. And we have inline oxygen sensors, so we can confirm we're below 0.5% oxygen levels. And with this, we've shown that we can maintain the complexity of human stool here. We take stool specimens, over 200 different types of bacterial species and strains from 11 different genera. And at the right is the density of microbes in between the villi. This is from a patient sample, and we can keep them alive for days, and you actually get better barrier function if it's a healthy microbiome. Now, we've been with Gates Foundation, we've now been obtaining organoids from patients with malnutrition, or it's called environmental enteric dysfunction from Pakistan. And we've been comparing them to healthy chips, and we could now model the microenvironment, including malnutrition. We do nicotinamide and tryptophan deficiency. We could do microbiome, we could have pathogens, we could look at immune responses, but just high level responses that we're seeing. If we look at a healthy chip versus chip with cells from ED patients, we can see that ED patients have decreased absorption of fatty acids and fluorescence here. And when we do nutritional deficiency, it's much worse. But more interestingly, as we've done transcriptomic analysis of these, and our collaborators in Pakistan have data from patients, and in the transcriptome at the right, you can see that if we compare the EED cells versus the healthy chips alone, normal media, rich media, they don't look like in vivo. If you do a healthy chip with -T, nutritional deficiency, it still doesn't look like it. But as you have these patient chips with nutritional deficiency, it's beginning to look closer, significantly closer to what we see in vivo, and we have not added the microbiome yet. So again, this now can be used to study really complex disease phenotypes. And so in addition to what I told you, we've found other key goals that we're interested in, which is predicting human responses to drugs using clinically relevant dose exposures, not bathing them, but what you see in patients, to develop personalized disease models, to create models that replicate immune responses that are complex, and to be able to replicate species-specific responses for that One Health challenge. We published a human bone marrow chip where we take CD34 and bone marrow-derived stromal cells from patients, mix them in a gel in one channel, endothelium in the other. We get normal white and red blood cell maturation over a month. But what we were able to show is that if we infuse drug, in this case 5-fluorouracil, a cancer drug, at the same perfusion or infusion as in patients at a clinically relevant dose of 4 micromolar over 2 days, we can mimic the toxicity seen in patients where you don't see this in static gels or suspension culture. People have seen this toxicity in vitro, but they bathe the cell in high concentration for many days. The dots at the right have different color of different patients, and that shows you patient-to-patient variation, which in this study was really minimal. AstraZeneca came to us where they had measured differences in toxicity depending on dose regimen, 2 hours over 2 days in patients. That's shown in gray at the top. So this is the pharmacokinetics, the PK, and we can mimic that on chip, and we mimic toxicities that are different between these two for the first time. This is real clinical mimicry. We've also made personalized chips with cells, CD34 from children with Shwachman-Diamond syndrome, have abnormal blood cell formation. We mimic that on chip, and we've even discovered that there are 2 subpopulations to these patients never seen before using these chips. And then finally, we are close to publishing a model of a human lymphoid follicle that you can literally vaccinate with a commercial flu zone vaccine, influenza vaccine, and you can see germinal center formation, plasma cell formation, specific IgG formation, and even cytokines produced that come in out the effluent that look like what is seen in blood of patients that are immunized. And we're now working with Gates Foundation and drug companies looking at vaccine development and adjuvant development. And again, this is the potential to replace non-human primates. And finally, with Emulate, we've done species-specific chips for liver toxicity, and we can show that we can mimic human, dog, and rat liver toxicities, which is very important because drug companies have to do preclinical tests, and dog and rat often get conflicting results. And I will just add that Emulate, it's really largely work of Emulate and Lorna Ewert, are about to submit a paper where they've now done almost 800 liver chips using 27 drugs that the IQ consortium has identified for proof of principle of the value, predictive value of these. And the results, I will just say, are extremely promising and better than anything that's out there right now. So I will end by just saying that we've developed many different chips. We've linked them together to do create human body on chips because we have a vascular channel line by endothelium. This is all published. We can quantitatively predict human pharmacokinetics based on computational modeling with data from chips. And the summary is that we really think that these are now ready to replace some animal models in the research labs in the drug development pipeline. It'll be one model at a time, but I really believe it's going to happen. And just say that as a Harvard faculty, I have to disclose that I'm a member of the board, chair of the SAB and hold equity in Emulate. 2010, we had tubes and pumps, and now you can buy these and it's automated system. And there are other companies selling chips as well. And with that, I couldn't do it without my incredible multidisciplinary team. And thank you so much. I'm happy to answer questions if we have any time.
Thank you so much, Donald, for this great talk. We will now move to our first short talk session, which is on MPS hardware and enabling technologies. I am happy to introduce our first speaker in this session, and this is Melissa Mendoza from Binghamton University. Melissa, we are looking forward to your talk.
Morning, everybody. I'm going to share my screen. So good morning from New York. My name is Melissa Mendoza. I'm a PhD candidate at Binghamton University, and I work under Dr. Gretchen Mahler. Today, I'll be giving you a small glimpse of the work that we're doing to develop an early and late calcific aortic valve disease on a chip. So first, I'll provide some background. So the aortic valve is one of your 4 major valves located in your heart, and the aortic valve specifically is situated between the left ventricle and the aorta, allowing the passage of oxygenated blood out throughout the rest of your body. And what's really fascinating about them is that they're really complex. So as you see here indicated by the numbers and the arrows in the images above, these are porcine leaflets, which are the tissue units that make up the aortic valve, resuspended in saline. And what you see here is that they're really thin, translucent pieces of tissue. But in vivo, they undergo many mechanical forces, laminar shear, oscillatory shear, and tensile stretching of the tissue. But if we dig even deeper into the microstructure of the leaflets themselves, we see that they're also very complex. So the leaflets are made up of 3 different layers: the fibrosa, the spongiosa, and the ventricularis. And these are mainly characterized by their extracellular matrix and are mainly composed of valvular interstitial cells. We also have valvular endothelial cells that line the aortic and the ventricular wall. For the purpose of the research that we're doing and the motivation behind this, we'll be focusing on the fibrosa layer, given that this is the location at which disease and calcification occurs. But it's also important to know that the spongiosa is made up of these glycosaminoglycans, or GAGs, that live in the loose connective tissue. And in the presence of disease, they actually end up migrating and depositing around those nodules in the fibrosa, which leads me to our overall motivation, which is to look at calcific aortic valve disease, or CAVD. And this has been described as a very active pathobiological process where we go from aortic sclerosis all the way to severe aortic stenosis. And as you see indicated by the arrows in the images at the bottom, it's largely characterized by the formation of these nodules, again, in the fibrosa. But literature has kind of divided this active process into 2 different stages. So the first is early CAVD, which has been described by lipoprotein deposition and oxidation, the infiltration of immune cells, and of course, the reorganization of the extracellular matrix. Whereas late CAVD has been largely characterized by the disorganization of the collagen, which is our predominant protein here, the deposition of those proteoglycans, again, that migration to the fibrosa and depositing around those nodules, and then, of course, increased leaflet stiffness and thereby inhibiting the blood flow, and having patients with aortic stenosis undergo invasive surgeries for total valve replacement. So this leads me to our overall objectives for this work, which is to create in vitro 3D microfluidic models of aortic valve fibrosa in both healthy and disease states, so we can further understand how this disease is progressing and hopefully be able to provide a test bed for new therapeutics. So to do that, we proposed 3 different models: healthy, early, and late CAVD. And first, I'll go over our late CAVD models, which is currently under review for publication. So to do this, we utilize primary cell culture, where we have porcine aortic valve interstitial cells embedded into a collagen 1 matrix, and porcine aortic valve endothelial cells seated over the top of that matrix to simulate the fibrosa. We also introduce varying concentrations of chondroitin sulfate as our choice of glycosaminoglycans, again, simulating that migration into the fibrosa and depositing around those nodules. And then we apply physiologically relevant shear stresses utilizing a peristaltic pump. So as you see here at the bottom, these are some images of our microfluidic device, which has been well published in previous studies in our lab. And we utilize primary multi-layer PDMS microfluidics. And so we place our dynamic and our static cultures into an incubator for up to 14 days and even 21 days. So I'll show you that. After which we assess our disease progression markers using alizarin red S or ARS stain, which is a highly used serological stain to detect calcium deposition in a cell culture. And then we also use scanning electron microscopy or SEM paired with energy dispersive X-ray spectroscopy or EDX to further analyze anything that may be forming as a result of the culture condition. So first I'll show you our ARS results. These are of course our qualitative images. So we see here when we have interstitial cells only embedded into the matrix, and then when co-cultured with endothelial cells, and going down the rows, we see an increase in chondroitin sulfate concentration. But of course, I'll point your attention to our high shear condition in our co-culture system, where we see a dramatic increase in the ARS detection. And we've seen this both qualitatively and quantitatively. But I'll point you to some more interesting results where we looked deeper into what might be occurring in this culture with SEM. So I'll remind you what that ARS image looked like for a co-culture with high chondroitin sulfate and high shear rates. And we create these beautiful mineralization structures. But of course, we wanted to analyze, what are these composed of? So EDX showed us the elemental breakdown of those potential calcifications that could be forming. And we're really excited to see that, of course, we have the presence of calcium and phosphorus deposition. But again, we wanted to formally calculate, what that mineralization looks like and compare it to the current literature. And we see that our CA to P ratios, specifically for our high shear rates, lie between a CA to P ratio of about 1 to 1.4. So for us, this was pretty exciting because if we compare to the current studies of ex vivo calcified aortic valves, we see that similarly to that, we are able to create a mixture of calcium phosphate phases. So monocalcium, dicalcium, and octocalcium phosphates are precursors to that of the creation of hydroxyapatites. So we did not see hydroxyapatite formation in our cultures, but we thought about ways in which we could potentially lead to that. So one of those ways was potentially looking at creating a long-term culture assessment. So here I'm showing you comparisons between 14 days and 21 days in our microfluidic system. And we see our SEM images as well as our EDX outputs. And what's interesting is that we found that the calcium and phosphorus elemental percentages were actually much higher at 21 days than at 14 days. But if we further calculate the calcium to phosphorus ratios, we find that we have equal or comparable maturity across our chip in as early as 7 days. So although we were not able to fully form hydroxyapatites in our chip, we are showing very promising results. Again, we do not use external factors such as oxygenic medium or even signaling molecules such as TGF-beta. So for us, this was pretty exciting. But now I'll switch focus a little bit onto the work that we're currently doing in the lab, which is to develop an early stage CAVD model using oxidative low density lipoproteins. So again, we have a very similar setup where we have interstitial cells embedded in that matrix, endothelial cells over the top of that matrix, and we introduce varying concentrations of oxidative low density lipoproteins or OXLDLs. And these concentrations are based on the literature as well as previous preliminary work that we've done in the lab. And again, we apply shear rates at high shear at 29 per centimeter squared, given that that's what the response was most dramatic at our 14-day studies. So then dynamic and static controls are placed into an incubator for 2 days. And after which we assess disease progression markers, again, using SEM-EDX. And we're also looking at alkaline phosphatase activity and also sulfated GAG production as using ALP, as using Alcium blue. And these are early markers of disease progression in CAVD. And we then normalize those values to the Bradford protein assay. So here I'm showing you our SEM images. So as you see down the rows, we're increasing our OX-LDL concentration. And again, I'll point your attention to our high shear rates in our co-culture. And you could see that there is a steady increase in the number of little nodules forming in our culture. But again, we wanted to further analyze that using EDX. So I'll remind you what that image looked like. And if we break down to our elemental compositions, we see again that we do find calcium and phosphorus in just 2 days. And what's interesting is that although this is preliminary data, we're pretty excited to see that our co-culture models are actually showing much higher elemental percentages compared to interstitial cell only cultures. And again, if we take that a step further to calculate the calcium to phosphorus ratios, our preliminary findings are actually showing that we're pushing towards hydroxyapatite formation. So for us, this is really, really exciting, although preliminary. We're excited to see where this goes. Then we looked at alkaline phosphatase activity, or ALP, and as we look at the literature, we know that increased ALP activity has been shown in the presence of CAVD. And we've shown that there are statistically significant differences between our static models compared to our dynamic models, where we see much more ALP activity in our dynamic system, which is pretty exciting. And currently the only results we have are for low concentrations of OX-LDLs, which hasn't really shown much of a difference compared to no OX-LDL presence. But we're hoping that future studies could show us something different. It may just be a small concentration that's not doing much of a difference. And then we looked at Alcium blue to detect sulfated GAP production and the recirculating culture medium of the devices, as well as the static models. And if, again, we look into the literature, we know that OX-LDLs are have shown to increase interstitial cell deposition of sulfated GAGs and overall promote the disease progression. And again, we see that in our dynamic studies. We're showing an increase of Alcium blue detection or sulfated GAGs production in our cultures compared to our static controls. So overall, we've been able to show you that we can develop this multi-layered device capable of recreating calcific aortic valve disease on a chip. And we've shown, although I couldn't show you today too much about our 21-day studies, hopefully you can see it in our publication. We've been able to see that we have viable 3D cell culture for up to 21 days. And in our late stage models, I've shown you that most of our dynamic calcification is mainly driven by the presence of high shear rates and the presence of endothelial cells. And we were really excited to see that our calcification formed in these microfluidic systems are comparable to that of ex vivo human calcified tissue. In our early stage models, although preliminary, we're really excited to see that in just 2 days, we were able to form high calcium phosphate maturity. And in the future, we hope to further incorporate immune cells, again, simulating that immune response. And of course, overall, we are in early development, but our big goal is to hopefully provide a test bed for new therapeutics. So with that, I'd love to thank you all for your attention. Specifically thank the MPS Scientific Advisory Committee for allowing me to speak to you today. These are our funding sources. Again, we are the Mahler Lab at Binghamton University, and I'd love to connect with all of you on LinkedIn. Thank you.
Thank you, Melissa. Very great presentation. And thank you for bringing us back on time. I have a quick question. So you mentioned for development, you using your chip model to develop therapeutics. So can you elaborate a little bit more on that? So how far you are in developing that, and how high throughput you can see that, and so what is your goal on that?
Yeah, great question. So right now, again, we are in very early development stages. We can run about 8 to 9 chips simultaneously. So we have really good high throughput. It's something we've worked towards pretty hard in the last couple of years. Right now, we haven't tested any drug therapies yet, but it's something that we're definitely working towards in the future.
Okay, thanks also from my side, Melissa, for the great talk. We now have Christopher McAleer from Hesperos as the next speaker. Christopher, the floor is yours.
Thank you. I appreciate you guys inviting me to give this talk. So I'm going to talk about a multi-organ system that we created for the evaluation of efficacy and off-target toxicity for anti-cancer therapeutics. Data that was published in Science Translational Medicine. And so being that this is a talk about MPS hardware, I wanted to start off with the design of our housing system. And so there are many different ways to design an MPS device, right? Some people like to use thermoform plastics. Some people will use PDMS injection molding, right? Many ideas about creating the housing system and then figuring out how to embed the cells into the housing system that's been created. We take a little bit more modular approach where, because we specialize in, and Hesperos in general specializes in multi-organ systems linking multiple organs in the same microfluidic stream, and the complications with doing that in the proliferation and maturation of different cell types, coming to the maturity, we like to create the cells and then build the system around them. And so, one of the things that you want to take into consideration whenever you're making an MPS device is what is the housing material that you're going to use, right? What is the design of the housing system that you're going to create? And how does that affect your ability to, one, the interaction of any of your drugs and cells with the platform itself? And also, how is that going to affect your ability to get any of the particular readouts that you're wanting, right? And so, in this particular case, Hesperos designs its own chips, right? Our own cantilever and microelectrode arrays, right? And so, we look at, cardiac propagation from electrode to electrode. We look at mechanical force output. And one of the things that we're very interested in is not necessarily just looking at toxicity of cells, right? Biomarker release, but, looking at function of the cells. And I'll show you that a little bit later. And so we made 2 configurations, and this work was supported by Roche Pharmaceuticals. So I'd like to put that out there now. The first one was to look at a very simple liver and bone marrow system, and then use a slightly altered design of that housing system to look at solid tumors and off-target cardiac and liver toxicity. And so, I want to make a point that also in the hardware that you're going to use, in the initial design of the housing system that we had made, there were some issues with our PCB boards, right, and the connectors that were there. And while we could measure conduction velocity and cardiac force and measure beat frequency over time, right, the signal to noise ratio initially wouldn't allow us to measure QT interval, right, or the field potential duration in vitro surrogate of the QT. And so, we have since fixed that, right, and reduced that. So, the complications that you get for both the housing system and its design, and also all the hardware that you use to collect the data. Right now, our signal to noise ratio is up where we can see that T wave and measure the FPD, right? But we like to focus on the functional measurements of cells. And so the first thing that we wanted to make sure is that, we took MEGO1s and Kasumi1 cells, they're leukemia cell lines, embedded them in alginate hydrogel, make sure that, we can monitor their proliferation in a linear phase, which we could over 14 days. And so we tested both imatinib, it's a tyrosine kinase inhibitor used to treat, it's a first generation tyrosine kinase inhibitor, and diclofenac. And while diclofenac is a non-steroidal anti-inflammatory, it's been used in adjuvant therapies with a lot of cancer, and actually shown on its own to have some cytostatic effect on cancer. And so, in our housing system, you can see that the control proliferates over time, both the MEGO1 and the Kasumi1, and that with imatinib and diclofenac, not only do you have a cytostatic response, but you actually have a cytotoxic response over time. And so then we also wanted to look at what do those 2 drugs do to the liver function in this particular case. right? And imatinib really showed no toxicity at all. And surprising to us, and it's still on my list of things that I need to go figure out why, we saw an upregulation of CYP2C9 and CYP3A4 activity, which is not necessarily clinically shown, but we did see decreased liver viability, which is known in literature, and a lot of that is due to diclofenac metabolism and intermediates that are made in the adducts that they form with the liver. And so it was also an indication that our liver was metabolically competent inside the housing system. And so from that, we were challenged to take it a step further from Roche and build a solid tumor to really look directly at an in vitro housing system, an MPS device, to measure metabolism of drug compounds, combinatorial therapies, the efficacy on solid tumors, the MCF7 and SW962, and the off-target cardiac toxicity, right? So, the MCF7 is a ER positive, MDR negative, right? We chose the SW962 because it's ER positive. So both of those should respond to tamoxifen or its metabolites, endoxifen, hydroxytamoxifen, right? But with the MDR positive, the PGP pump, right? The efficacy of drug compounds is limited, right? Unless you can shut down that PGP pump. And so we specifically chose verapamil as the first generation PGP inhibitor, mostly because we wanted it for its calcium handling issues, right? And we expected that to cause cardiac toxicity. And so this is a little bit busy of a slide, but so we looked at the efficacy of tamoxifen in our MCF7 and remind you that these are MDR negative. So tamoxifen, without a liver really doesn't do much. And it's known that endoxifen or hydroxytamoxifen is somewhere between 10 and 100 times more effective than tamoxifen alone, right? If we add a liver compartment in there, which is the beauty of the way that we design these, we can create the same exact system and just, in parallel, leave out a liver component. And so with a liver and the metabolic conversion of tamoxifen to its metabolite, right, we see a statistically significant decrease in the viability of the cancer tumor, right? And if we just add the same concentration of hydroxytamoxifen, right, we get a slightly, while not statistically significant increase, which for me indicates that our liver is pretty highly metabolically competent if we can use similar concentrations and get similar results in terms of its efficacy. And then, we added verapamil, PGP inhibitor, and while there is no MDR pump, it should have no effect, which we show that it did not. Right, but when we look at the SW962, right, which has a PGP positive, right, so when the drug gets absorbed into the cell, it gets shunted right back out. And so whether you use tamoxifen with a liver, without a liver, or the hydroxytamoxifen, there is no, we showed that there is no effect on the cancer viability. But when we use a combinatorial therapy of tamoxifen in the presence of the liver with verapamil to shut down the PGP, we get a statistically significant decline in the cancer viability. And so that was really promising for us. And so we also, as a positive control, wanted to make sure that that verapamil addition in the SW wasn't actually causing the toxicity. And so, in this case, we used 300 nanomolar in the previous results, and there is no effect of verapamil alone on cancer viability. And so, simultaneously, while we were measuring the viability of the solid tumors, we wanted to look at the effect on cardiac function. And, as I'm sure a lot of people are aware that anti-cancer therapeutics are known to have cardiac damage and even long-term fibrosis and necrosis. So looking at our control systems, carrier dose controls, right, there is no effect on conduction velocity or force or beat frequency. But when we add tamoxifen alone, right, for both the conduction velocity and the cardiac force, right, we get a decline in the first 24 hours, right? And due to the interaction of tamoxifen with the housing system, metabolism of tamoxifen, right, and the fact that we feed them daily 30% over the course of 3 days, right, as the tamoxifen is removed, we get recovery with the tamoxifen alone, right, for both conduction velocity and force, right? But in the combinatorial therapy where tamoxifen is associated with calcium handling, sodium and calcium handling issues, and verapamil being associated with calcium handling misregulation, right? In the presence of the combinatorial therapy, we get a further decline than we have in tamoxifen alone, and it doesn't recover over time, right? And I want to point out that that's not due to the death of the cells, because if you look in panel D, we look at both tamoxifen and the combinatorial therapy of tamoxifen and verapamil, and there is no statistically significant decline in the viability, right? But interestingly, I didn't expect to see this result when we first looked at it, and I said, I don't understand why the three of these don't jive, right? When you look at tamoxifen declines and comes back, and the combinatorial therapy declines and it recovers, right, for beat frequency. And I think that has something to do with the way that the cardiac function goes in our housing systems, right? For both conduction velocity and force, an intact syncytium is necessary, right? And so I'm not entirely sure if, the beat frequency, not needing a very well organized syncytium, is a little more robust in its ability to handle the calcium handling problems, but that's pure speculation at this point. And so the other thing that I wanted to talk about was when you design your housing system, right? So we designed ours, we use COMSOL and CFD-ACE modeling, right, to look at fluid retention times in individual compartments, to look at shear stresses on the cells in any department, because for some cases it matters, and in other cases, for some cases it matters greatly, and for other organ types, not so much. Right. And so you want to look at that. You also want to eventually, when you take the pharmacodynamic data that you're collecting, right, the drug effect on all the cells, you want to eventually put that into a PK/PD model. So, looking at, measuring the HPLC content of the drug. So even though we gave a 10 micromolar bolus dose of tamoxifen, the maximum concentration, in the system only came out to be about 7.5 micromolar. And due to interactions that occur, both metabolism and interaction with the housing system itself, right, we measure that concentration over time, and we can both look at maximum concentration driven effects and area under the curve effects. And so it's really important too, when you look at drug dosing inside the microphysiological system, to take into account any interactions that your drug is going to have with the MPS device, right? And I'm sure, everyone here is aware of the issues with lipophilic compounds with PDMS, right? But, I'm hoping in the meeting in New Orleans that, we'll be able to present some data on a litany of drug compounds that span the lipophilic profile and how they interact with different housing materials like polymethyl methacrylate and even like tissue culture treated polystyrene, which indicates, our preliminary data indicates that you might even get 30% loss of some lipophilic compounds just to polystyrene, right? So it's really important to take into consideration the concentration of the drug measured directly by HPLC, right? And whether or not, one other consideration is, the free drug concentration versus the total drug is, our system uses serum-free medium. That being said, there is quite a bit of albumin in it, right? And so there is plasma protein binding of the drug, right? And looking at what's available to the cells versus, the total drug concentration. And so, we, use all these things to then, put into models for predicting clinical trial data or human in vivo data, using PK/PD. And so I just wanted to use this paper that we had published, a bit ago to talk about the design of the housing systems and how the materials that you use, the conformation of the system are important in the outcome and the feasibility of your experiments and what readouts you can actually get. To our knowledge, it was the first real MPS system capable of monitoring efficacy and off-target toxicity. There have been some that have come out since then, right? And we really think that these MPS devices and the way that our systems are interlinked give us the real potential to measure therapeutic index in early stage development, right? And so when you start to look at efficacy, a lot of drugs, the real efficacy in humans doesn't come till phase 3 clinical trials, where quite a few of them, I'd say a vast majority of them fail, right? So if we could really look at human relevant efficacy in early stage development, I think we can increase the success of drug compounds and moreover, I think decrease the failure rate of compounds. And for us, the open system design allows for reconfigurability and selective removal of organs to look at what one particular organ effect is on the rest, right? Say, rhabdomyolysis, removing skeletal muscle and seeing if you still have kidney damage, as an example. And so that's my talk, and here's my information if you need to contact me.
Thank you, Christopher. Very interesting presentation. I have a couple of questions to you. So my first question is to the cell sources you selected for the tumors. So what is your rationale to go to the cell lines, immortalized cell lines? I understand that probably primary cultures, that's hard to obtain, but there are certain issues about using immortalized cell lines, especially MCS7 is has genetic unstable, and so there are several concerns about that. So what is your perspective on that, and whether you are planning to move to patient-derived tumors probably?
Yes, for sure. And so the original thought behind that was, and I understand the considerations with MCF7, we really only chose it for its ER positive effect. And so the initial cell lines were used as a proof of concept, right? To show that, we could build the housing system, that we could get the efficacy and off-target toxicity that we wanted, right? And those cell lines are easy to work with, and they're pretty well established. The plan moving forward is to, ultimately we would like to transition this system to tumoroids that we get from patients, right? And look at how, ultimately we think the system can be used for personalized medicines to take the tumoroid and take them in the housing system and look at, a litany of compounds, both, looking at the, what is the word I want to say? The histology of the tumor that's taken out originally, right? And use that data to inform, what arsenal of drugs and combinatorial therapies we would want to use on an individual patient tumor. And so, we're doing some ongoing discussions and work on that, but that's proprietary at this particular point.
Yeah, that's fine. Another question I have is, you have for now cardiac and liver, so another organ of off-target toxicity, which is of concern, would be kidney. So is any plans to add the kidney organ on chip to your body on a chip platform?
Yeah, I mean, so if you go to the Hesperos website, you'll see that we have quite a few organs beyond just the liver and cardiac and cancer. We do have a kidney module, both a glomerular component and the renal proximal tubule, to look at, kidney damage. And at this particular point, the kidney module that we have does not have an elimination function. It really is just a toxicity target. So we can look at like glomerular clogging or, toxicity to the RPTCs, right? Look at, tier changes and KIM1 release and the rest of it, right? But at this point, we're not quite far enough along in the kidney to use it. The ultimate goal was to use it both as a toxicity target and filtration and elimination, but we're not quite there yet.
Okay, thank you. I have one more question on chat from Abhinav Bhushan. Very interesting. In other words, what is the key market segment for Hesperos? How big is the market in dollars for MPS devices that Hesperos has?
So you can talk to Dr. Hickman, but I think, where Hesperos wants to be is, I think, we don't want to do early stage, like high throughput screening. That's not really what we do. What we're really looking at is, very targeted high content screening. And so what we think our market is going to be is in rare diseases. Right. We pride ourselves on being able to create the system that you want, right? We'll try and generate a phenotype and then, test the compounds against it, and so at this particular point, actually looking at direct interaction of cells in a common microfluidic stream is something that only Hesperos can do, to my knowledge, right? Because we have developed a proprietary serum-free medium that allows for the function and viability of cells to maintain over up to 28 days. And so I think that looking at the cell-cell interaction in those and rare diseases is where we really want to be.
Great. So one more question from Pelin. Hi, Christopher, very interesting talk. Are you seeing any potential to use this model for cell therapy use, specifically with testing of tissue tox or pharmacokinetic, pharmacodynamic?
And so, we are working with quite a few partners to feed their PK/PD algorithms. So absolutely, one of the things that we pride ourselves on is measuring the pharmacokinetic profile and both efficacy in pharmacodynamics and toxicity in the pharmacodynamics. And I'm sorry, I don't know what the beginning of that question was other than the PK/PD.
Whether you're planning to use the cell therapy for cell therapy use.
Well, I'm not completely sure what you mean by cell therapy use, but if we're talking about biologics, sure, we already do that. We have to be clear about the positives and the negatives, right? What your system can and can't do, right? And so, like in that particular cardiac system that we had created there, if you're looking at, like, say, cardiac fibrosis, right, or inflammation of the perineum, right, that system is not quite prepared to do that, right? And so we are in the process of building and designing and building a system that looks at a three-dimensional cardiac tissue, right, that will start to look at inflammation and fibrosis, right? But they'll have to, at that particular point, they would be parallel therapies or parallel systems that would be run to give a totality of the picture of what happens in the cardiac damage, right? Because when you start to look at like three-dimensional models, like in that particular domain, right, some of the function, right, like we don't see a way that you can measure like force output, which we hopefully at some point can directly correlate to ejection fraction or the conduction block of the cells, right, in that model.
Thank you. You have one more question. Yeah, and you have one more question. Please answer that in writing on the control of the flow in your system, and we will move forward with our agenda. Thank you.
Sure. And just in case I didn't say it, I would like to thank Roche Pharmaceuticals for funding that work.
Thanks.
Thanks to Christopher for presenting. Our next speaker is James Tronolone from Texas A&M University, and he's one of our 4 nominees for the Young Investigator Award. So welcome, James.
So good morning. Today I'll be detailing my work using machine learning to optimize microvasculature on a chip in support of generating next generation vascularized organ chips. I'd also like to highlight that this work is part of a range of projects aimed at generating translatable organ chips in Dr. Abhishek Jain's lab at Texas A&M University. So as I'm sure most of you are familiar with, the body or microvasculature is a key component of many tissues in the body. In fact, vasculature can be considered one of the largest organs of the body in terms of surface area. Blood vessels are responsible for oxygenating organs such as the brain, digestive tissues such as the pancreatic islets and the villi of the small intestine, as well as filtration organs such as the kidneys. So much work has been dedicated towards generating microvascular networks on chip. And recently some reports are emerging detailing microvascular networks co-cultured with tissue-specific cells. And this marks an advancement of the MPS field. My work specifically involves developing models of vasculogenesis on a chip for translation to pancreatic islet models. To accomplish this, I use a microfluidic device which contains 5 parallel microchannels, and each microchannel is separated by a semi-permeable barrier that's formed by patterned PDMS coats. When I load biomaterials into the chips, I'll inject a viscous hydrogel prepolymer solution, cross-link that gel to form a semi-solid material, and then I inject fluids adjacent to the hydrogel channels, thus creating an alternating hydrogel fluid arrangement, which later facilitates 3D cell culture, as well as crosstalk between the channels. In such a chip, I can encapsulate growth factor secreting cells, which form a growth factor gradient towards a central channel housing an individual or a suspension of individual endothelial cells. And over a set culturing period, these individual endothelial cells will eventually grow into perfusible microvessels, such as the ones shown in this fluorescence micrograph here. So to demonstrate a working chip, I'll show a typical vasculogenesis chip arrangement where lung fibroblasts are situated in the outermost channels and secrete proangiogenic factors towards the central channel containing endothelial cells. In this video, you can see that eventually the endothelial cells will self-assemble into capillary networks. And then additionally, these networks can be perfused with fluorescent microbeads, demonstrating their functionality. So indeed, much work has been dedicated towards developing vascularized networks on chip in our own lab and many others. And we actually recently published a review article in Advanced Materials summarizing the past 10 years of vasculature on a chip examples, where we found that there was actually a 5-factor paradigm contributing to all these different examples. This paradigm included growth factors, mechanical forces such as shear stress or interstitial flow, different biomaterial configurations, endothelial cell density or source, and supporting or pro-angiogenic cell source type and density. So all of these contribute to different examples of angiogenesis, lymphangiogenesis, as well as vasculogenesis on a chip. Yet, when we look to optimize the networks based on this paradigm in our own lab, it became unclear what parameters of a network we should optimize based on the traditional tools at our disposal. For example, we have an experiment here where we tested different matrix stiffnesses by increasing the concentration of fibrin in the hydrogels. And for some metrics, the 5 mg/ml condition is significantly higher than others, while the same can be said about the 2.5 mg/ml condition when looking at different metrics. So for example, when we look at vessel length, 5 mg/ml group has significantly higher vessels, but the 2.5 mg/ml group has significantly larger segment or vessel segments, as well as wider vessel segments. So in response to this, we reflected and determined that maybe there's an alternate metric we ought to optimize. Because if you think about it, the morphology might not be as important as long as it accomplishes the role of a microvascular network. And that role is the transport of different species, so oxygen, nutrients, waste, or even drugs. So we devised a metric to quantify the perfusibility of a network by using numerical analysis. And when running numerical analysis, we apply an inlet concentration and measure how much of that concentration ends up at the outlet. And we quantify that in a metric we call the normalized mass transport. However, there's a major downside of running a numerical analysis such as this on larger data sets, and that's that there is a large computational power and time needed, which in turn severely limits the translatability of this new metric. So we wondered whether we can use a machine learned regression model that might allow us to determine an equation between the easy to obtain morphological metrics here and relate that to our simulated mass transport, which is the function of the networks. In doing so, we aim to develop an object containing an equation that can be used in data analysis software such as Python or MATLAB. And an object like this could be easily downloaded and utilized by other researchers without the need for high computing hardware. So in developing a machine learning model, we decided to use our neural network regression model. And to build this model, we took 500 samples of different network morphologies and trained a machine using 80% of that data. And of that 80% of the data, 20% was used for 10-fold cross-validation, which is a typical validation method used in machine learning. So after a series of trainings, we determined an optimal neural network architecture that took in 6 morphological metrics, combined each into 128 nodes of a first layer, the outputs of which fed into another layer containing 32 nodes. And finally, the outputs of the second hidden layer were combined to form an output layer. The output of the network at the output layer is a predictive metric that we call the oxygenation metric, which theoretically is highly correlated with the actual simulated values we obtained through our numerical analysis. So further, I'd like to also highlight that while this might look like a complex model, neural networks can be simplified as a very large multiple regression equation, where the machine is learning what weight to add from each node, what bias to add to each node, what activation function to add to each node. And then at the very end of the model, we have the summation of all these different functions. So it functions similar to a multiple regression model where we have an output defined as many different coefficients and functions that are dependent on the original input layers, or the original input morphological metrics. So after the machine learning model is trained, we can assess its accuracy on the testing data set. And here we can see that it generally follows a trend of expected values, and we can obtain an accuracy metric called the mean absolute error. And in this test, the mean absolute error was 0.09, meaning that our model averaged a 9% error compared to the true value or the simulated value. When evaluating the difference between expected and predicted, or in other words, when comparing the simulated values with our oxygenation metric, that's the output of the neural network model, you can see that most values are close to or at 0. Further, we can see that for this data set, the oxygenation metric is most correlated with the simulated mass transport, showing that it outperforms the traditionally reported vascular networks, or the traditionally reported vascular network metrics. Another advantage of using a neural network is that we can employ Shapley additive explanations, which is a recently published game theory algorithm that allows us to determine which neural network input contributes most significantly to the predicted output or oxygenation metric. For the entire data set, we can see that branch point count and vessel coverage have the highest impact on the SHAP value, which is interesting because most of the previous literature will actually present vessel length and diameter, and some will report vessel coverage, but branch point count is rarely reported in previous examples of vasculogenesis on a chip. Further, we can see that branch point count and vessel length correlate highly with the SHAP value. As each of these increase, the SHAP value increases. Further, we don't see the same positive correlation when looking at vessel length or segment count, which, if you remember, is the most reported of the morphological metrics. So for these two, sometimes high values will give high SHAP values, but also low values can give high SHAP values. And the same can be said about the segment count. So we ran these SHAP analyses for all of our 500 samples. But for the sake of time, I'll just only discuss the same hydrogel stiffness experiment I mentioned earlier. And we can see that for samples in this experiment, the average mass transport for the average oxygenation metric was 0.53. So 53% of an initial concentration makes it to the outlet of the network. For the least stiff network, we see that the vasculature has elevated mean segment length, vessel coverage, and mean segment diameter, all of which add a combined 37 points or 37% to the oxygenation metric. So we have a 37-point increase in the metric due to these morphological traits of this specific network. On the other hand, a 5 mg per mil network has a similar oxygenation metric, but is optimized chiefly due to a higher branch point count followed by vessel coverage. These two combined increase the oxygenation metric by 31 points. So using a SHAP analysis, we can also dissect how each experimental group's morphological metrics impact the oxygenation metric. Additionally, we can see that for the two highest stiffness groups, each have oxygenation metrics close to 0. For each condition, low vessel coverage values decrease the oxygenation metric by 16 and 12 points for the 7.5 and 10 mg per mil groups. And then mean segment diameter, mean segment length, and branch point count are the next three highest for each of the groups, all contributing to a loss about 20 points in the oxygenation metric. Thus, it becomes easy to see that any of the two lowest stiffness hydrogels could have a high oxygenation. Yet, as I mentioned earlier, the original metrics don't really show this, with each of the 6 except vessel coverage and branch point count showing significant differences between the 2.5 and 5 mg per mil groups. Further, we can see that the oxygenation metric is most highly correlated with mass transport in this specific experiment, and this matches that of the entire data set that I presented earlier. So the most important application of this tool and the whole motivation for running such a large machine learning study was so that we can translate microvascular networks to advanced MPSs. So in the next few slides, I'll detail how we decided to vascularize pancreatic islets because there's a growing need to develop cell therapies for type 1 diabetics who are eligible for islet transplantation. But the current market of islet transplantation therapy suffers from an inability to oxygenate the cell therapies, and this eventually leads to graft death and the return of the type 1 diabetes symptoms. So when recreating this system on a chip, we first expanded islets, endothelial cells, and fibroblasts, and further cultured islets on a spheroid formation plate in order to generate a more physiological phenotype. And then islets and endothelial cells were combined in a fibrin hydrogel and injected into a central hydrogel, or a central channel of the microfluidic chip. And then separately, lung fibroblasts were encapsulated in fibrin hydrogel and added to the outermost microfluidic channels. And then after a 96-hour culture, we generated microvascular networks that fenestrated the islets, similar to what you'd find in vivo. So as I mentioned earlier, hypoxia is a major limitation that islet transplantation therapy has to overcome. So in addressing this issue, we decided to challenge our vascularized islet chips with hypoxia to determine their functionality in a kind of mimic of islet transplantation. So after a 4-hour hypoxia challenge, we added fresh oxygenated media containing glucose for 1 hour. And then we collected the effluent media from the fluid channels, and then also digested the hydrogel and separated the digested hydrogel containing insulin from the cell pellet. We then measured the chip effluent and the hydrogel effluent together using an insulin ELISA. We found that chip formulations previously generating oxygenation metrics of 0.25, 0.5, and 1 increased the insulin secretion of the islets, with the two highest oxygenation metric groups increasing the islet's insulin secretion almost twofold. So such an increase in the physiological response of pancreatic islets, even under hypoxia challenge, validates the machine learning work that I previously presented, where we're trying to optimize the oxygenation or the biological function of the microvascular networks. So thus we showed that optimizing an oxygenation metric has physiological benefits in this model. And I also hypothesized that other systems could benefit from this optimization workflow. Having created an optimized vascularized islet chip, we also have a platform that can be used in two fields. The first is disease modeling, where we can further challenge islets with autoimmune cells, similar to the type 1 diabetes response. And if that's successful, we can use this as a testing bed for type 1 diabetes therapies. On the other hand, there are also potential applications in tissue engineering, where we might further look to optimize the oxygenation of pro-angiogenic biomaterials, pro-islet biomaterials, or even immunoprotective biomaterials in an effort to more effectively devise islet transplantation materials. Further optimizing such materials might reduce the number of animals needed before human clinical trials, as well as the overall cost of developing a new islet cell transplantation therapy. And I might be a little over time. I just want to quickly acknowledge my lab group, especially my advisor, Dr. Jane, and Tanmay Mathur, who helped me a lot with the numerical simulations, and Christopher Chafdari, who worked with me all of last year generating a lot of our samples. He graduated, but he now works at the FDA. I would also like to thank my family for their support, as well as the NSF and Texas A&M Biomedical Engineering Department for my funding, as well as Texas A&M for grants that funded the actual project. So with that, I'll be happy to take any questions.
Thank you, Jim. You have two questions in the chat. First is from Lawrence Vernetti. Do the islet spheroids vascularize or just become encapsulated by the microvascular network?
Yeah, so we co-culture at the first day. We have islet spheroids and endothelial cells. An individual suspension of endothelial cells surround the islet spheroids, and then over the course of 96 hours, the networks grow and eventually grow around, and some of them will actually fenestrate through islets. So I guess kind of to answer that question, it might be a little bit of both. Definitely the networks go around the islets, but also through some of them as well.
We have another question, and if you have a short answer, please speak. Otherwise, you should type it. It's a question from Almond Navas. Can you talk a little more about developing and writing the machine learning algorithm?
Yeah.
Is there a short answer to this question?
I mean, maybe briefly, and then maybe I could type more later, but the general workflow would be that you have a data set, then you go through and clean the data set to remove anything. It might be a little rude, but most people call it garbage data. So you remove garbage data that can affect the performance of your model. And then you separate the remaining cleaned data set into 80% for training and 20% for testing. And 80%, that training data we go through and just tune different parameters of the neural network in order to find what optimizes certain accuracy metrics, such as the mean absolute error, which I mentioned earlier. But you could also look at other metrics like the R-squared value or the mean squared error and things like that. But I'll also type more just because I've spent months developing machine
I'm not
learning.
removing the question. So we have 1 more minute for one question from Andres Dines. Are the islets more oxygenized with the endothelial capillaries around than just being in the flow?
Yeah, so that's a good question. So the way this assay works is we grow the chips, and then we put the chips in hypoxia for 4 hours to induce a hypoxia stress. And then we remove the media and add fresh media containing glucose, but that's also oxygenated media. So then that media flows through the capillary network and reaches the islets, hypothetically oxygenating them so they'll produce more insulin compared to something you'd see on the left here, maybe where there are no networks, so that oxygen doesn't really reach those islets, resulting in less insulin secretion.
Thank you. Okay, we move on, and then you answer the algorithm question.
Yeah, that sounds great.
Okay, thank you, James. We will now hear Kenta Shinha from Tokai University, also a nominee for our Young Investigator Award. Kenta, you have the floor.
So thank you for your introduction. So I'm Kenta Shinha from Tokai University in Japan. So today I'm talking about microphysiological system platform with high operability and pumping capability for commercialization. So breathing lung on tip is one of the best known MPS in the world. Research on MPS for reproducing such in vivo environment is very active. And the number of MPS-related papers is increasing every year. In recent years, the industrialization of MPS has been promoted mainly in the United States and EU. So many companies have been established to develop and sell MPS, and the market scale of MPS is expanding year by year. So in Japan, also it started rather than the US and EU, a project for the industrialization of MPS in underway since 2017. This project aims to develop MPS that can be applied to the evaluation of the safety and pharmacokinetics in the process of drug discovery. So collaboration between industry, government, and academic. So in this project, we developed 2 MPS with a focus on improving operability for commercialization. Today, I would like to introduce the 2 MPS. The first is the MPS, a conventional membrane type organ model with 2 channels separated by a porous membrane. So among the organs in the human body, the BBB, gut, and kidney are important organs. In these organs, substance flexibility permitted between tissue and blood vessels. Therefore, to evaluate the efficacy and toxicity of drug related to these organs in vitro, it is necessary to culture cell in a 2-layered channel. The many membrane type organ model MPS have been proposed. We aim to improve operability and throughput by developing a conventional organ model chip and fluidic system and observation system. First, the detail of the organ model tip. We have developed a tip called Fluid 3D Cross in collaboration with TOK. So Fluid 3D Cross is a conventional organ tip that has two channel layers, so separated by porous membrane. It is made of engineering plastic, which is characterized by low absorption of substance. So we show three experimental applications of Fluid 3D Cross. The first application is apply shear stress to cells. It is expected to become an organ model with high physiological function by reproducing the flow in vivo. The second is the co-culture of epitheal cell and endothelial cells. It is expected to improve physiological function by seeding cells on the top and bottom surface of porous membrane and constructing a 3D structure. The third is evaluation of substance transportation. It is possible to evaluate functions such as absorption and excretion by introducing reagents into either layer and evaluating mass transport to the other layers. Next, I will introduce the fluid platform of the membrane type organ model MPS. One of the problems for the commercialization of MPS is its lack of usability. In experiment using the MPS, it is necessary to connect the pump to the organ model chip with a tube. For example, the MPS shown in this picture cannot be used by many people because it is very complicated to handling. So therefore, we have developed a fluid platform to improve the usability of MPS. So we integrated all functions such as pump and power supply into MPS cassette, which is the same size as a well plate. The MPS cassette fabricated with a 3D printer has a medium reservoir and channel structure. So perfusion can be performed by placing a pump and the fluid 3D cross on the cassette. Therefore, with our MPS, many people can start experiment in a few minutes. So we performed a cell-based assay to evaluate the usefulness of the developed MPS. First, we applied a different shear stress to proximal tubule model cells and evaluate the gene expression level related to drug transporters. The result showed that the gene expression level increased with shear stress. In addition, we confirmed that the optimal shear stress was different depending on the gene to be expressed. Next, we confirmed drug toxicity tests of proximal tubule model cells. So cisplatin is an anti-cancer drug that causes nephrotoxicity. Cimetidine is an inhibitor of drug transporter. The results showed that the drug toxicity in the perfusion environment was lower than in the static environment. In addition, the inhibitory effect of cimetidine on nephrotoxicity was also higher in the perfusion environment. So these results suggested that shear stress sensitive cells should be assayed in a perfusion environment. In addition, we are working with Nikon to develop an automated cell dynamic observation system to improve the throughput of experiment. So this video shows the inside of the system. This system is based on Nikon's BioStation CT. BioStation CT is cell culture and observation system. We have optimized the MPS for the BioStation CT and installed a power supply system for the MPS in the BioStation. So the BioStation can perform both bright field and fluorescence observation. In addition, the BioStation is installed with volume contrast method, which is Nikon's original method. The Wyss method analyzes the Z-stack image in the bright field and recreated an image. This allows for the clear observation of cell on the membrane without staining. We analyze the Wyss image to evaluate the number and morphology of cell and the area of intercellular space. The result shows that changes in cell morphology caused by cisplatin toxicity could be observed more sensitivity than fluorescence image. In addition, we conducted a permeation test of lucifer yellow in the BioStation CT. So lucifer yellow is a substance for paracellular transport in cells. The result shows that the higher the concentration of cisplatin, the more nephrotoxicity occurred, and the higher the leakage of lucifer yellow. So we designed a membrane type organ model MPS optimized for BioStation CT and realized time-lapse observation of multiple MPS. So this achieved reduction in experiment load and increase in throughput. So for the 3D cross is now available from TOK. So secondly, I will introduce the Starler-based kinetic pump integrated to MPS for co-culture of multi-organ model cells. A number of organs interaction have been reported in vivo. So therefore, it is necessary to evaluate the pharmacokinetics of organ-to-organ interaction in drug discovery. So therefore, we developed a very simple multi-organ MPS to evaluate the interaction between various organs. The concept of our MPS is to use organ model as blocks that can be placed freely on the MPS platform. In this MPS, Starler-based micropump is integrated in the flow channel. So this pump is rotated by magnetic force and can perfuse the culture medium in the MPS. So this kinetic pump integrated microfluidic plate has been developed in collaboration with Sumitomo Bakelite. The culture chamber has the same design as 24-well plate. Therefore, the user can use the Kim plate with the same operation feeling as the well plate. So this graph shows the evaluation result of the flow rate in the Kim plate. Also, this graph shows the diffusion of lucifer yellow in the A chamber into the B chamber. So this result indicated that the flow rate in the Kim plate can be controlled by the rotation speed of the Starler-based kinetic pump. So we performed co-culture of intentional and hepatic model cell using Kim plate. In the iPS-driven intentional model cells, the TR and gene expression level related to tight junction are increased. On the other hand, in hepatocytes derived from chimeric mouse, co-culture tended to increase the metabolic function of the drug. So from this result, we conclude that intestinal and hepatic model cells enhance each other's functions by co-culture. So finally, we conducted a drug efficacy test using the template. We compared two methods, the conditioned medium method and co-culture on the MPS. In the conditioned medium method, the cancer model cells were cultured in the same medium used to culture the hepatocyte. So CPT11 metabolized by a hepatocyte to SN38. So this graph shows a survivability of MCF7 in the culture with a conditioned medium. The survivability was higher than the co-culture on the template, indicated that the drug effect was estimated to be lower. So from this result, we conclude that the accuracy of drug efficacy can be improved by consideration of the change in drug concentration due to metabolism. So the template will be soon available from Sumitomo Bakelite. So this page is our research summary. So we would like to special thanks all of them. Thank you.
Kenta, we will move forward and you can just type your answer in the Q&A box.
Okay, sorry. Thank you.
And we move forward with our program, which is BioBreak tomorrow.
Thank you, Kenta. So Kenta was the last speaker of our first short talk session, and a big thank you to all speakers so far for the great presentations. We will now have a very short break for 5 minutes, and after that we will continue with our second short talk session, which will be on cell models and applications for MPS. Ricardo Barril from the University of Cincinnati will move you through the second part of our event. So I would like to thank all of you for joining us so far, and we will be back at 11 a.m. EST.
And I get one more reminder, please follow the link to the poster book and look our amazing posters.
Welcome back after this short presentation. I'm Ricardo Barril, and I'm assistant professor at the biomedical department of the University of Cincinnati. I'm excited for this opportunity to co-chair the second part of today's meeting that focuses on cell models and application for microphysiological system. I will welcome all speakers, and as previously, Lina will conduct the Q&A session. I would like to kindly remind all speakers to stay on time and the audience to type any question in the Q&A tab. And now, with no further delays, I have the pleasure to welcome our first speaker of the session, Dr. Devin Mayer from the Johns Hopkins University, who is also one of the 4 nominees for the Young Investigator Award. Devin, the floor is yours.
Thank you. So one quick correction, I'm currently a PhD student here at Johns Hopkins University, but hopefully I'll be Dr. Mayer soon. Thank you for that. So my project, we're focusing on using a human-induced pluripotent stem cell-based 3D microphysiological system for modeling cardiac dysfunction and microgravity. So humanity is approaching long-duration spaceflight with NASA currently planning a mission to Mars. So the health effects of spaceflight on the cardiovascular system in particular, as well as of the rest of the body, are currently hot topics for research. And NASA has specifically identified several cardiovascular risks as areas of high priority for research, including diminished cardiac function, occurrence of serious cardiac dysrhythmias, impaired responses to stress and exercise. While we've gained important insights from prior studies using 2D models, a shift to 3D in vitro models is imperative in order to really characterize these effects. So the goal of this project was to develop a high throughput microphysiological system of cardiac muscle derived from human induced pluripotent stem cells to study the effects of microgravity. So this is our engineered hard tissue platform. It's made from induced pluripotent stem cell derived cardiomyocytes and stromal cells seated within a hydrogel scaffold, but then self-assembles between two silicon posts, which produce twitch forces that deflect the posts. And based on the deflection of the post, which you can see in the video here, we can calculate twitch forces as well as quantify a variety of other metrics of cardiac function. However, traditional engineered hard tissue models require microscopy to measure the deflection of that post, and astronaut time is extremely limited on the International Space Station. So in order to improve the amount of data that we could acquire, while also limiting the amount of astronaut requirements we had and resources we required, we further modified the engineered hard tissue platform by incorporating magnets into the flexible post. These magnets embedded in the post are then placed over a giant magnetoresistive sensor, which allows us to measure post deflection in an automated and real-time manner without the use of any astronaut resources whatsoever. So we could send these tissues up to the International Space Station, keep track of cardiac function over the month that they were up there, and have an idea of diminished cardiac function as a result of their time on the International Space Station. Another problem that has been a recurrent problem in the field is the relative immaturity of cardiovascular tissue, engineered tissues, due to the use of these induced pluripotent stem cells. So in order to further improve the maturity of our engineered heart tissues, we opted for a biomaterials approach. This was actually published this year in Biomaterials, where we isolated the left ventricle of the pig heart, decellularized it, and then mixed it with reduced graphene oxide to create an electroconductive hybrid hydrogel. And we, with these tissues, we compared to collagen, just decellularized extracellular matrix, and the decellularized extracellular matrix with reduced graphene oxide to improve the electroconductivity, as well as a DCM transglutaminase control. Because the reduced graphene oxide increases the stiffness of the tissue, we artificially increase it using that transglutaminase to make sure the effects we are seeing are due to the reduced graphene oxide and not due to the increased stiffness. And what we found, as you can see in this graph here, is that the twitch forces are improved significantly as in the DCM-RGO tissues for the duration of the experiment. We also saw increases in myosin heavy chain expression, as well as other markers of cardiac maturity, increased sarcomere lengths, and improved electrophysiological function. So with our improved tissues, we then started preparing for a spaceflight mission. So working with Bioserve Space Technologies, we developed this engineered hard tissue spaceflight chamber, where in space, media changes are problematic. You can't have free floating media because in the absence of strong gravitational forces, it's just going to float away. So we created this fully sealed, self-contained tissue chamber where we can have our tissues beneath this PDMS membrane that allows for gas exchanges with a hard polymer body that prevents any leaks, with ports on the side to allow for syringe-based media changes. So these chambers then were fit into a circuit board integrated plate habitat designed by Bioserve, where we have our tissue chambers here. As you can see, they're placed over these circuit boards that have the giant magnetoresistive sensors so that we can keep track of cardiovascular function, and placed into a fully sealed plate habitat for culture on board the International Space Station. So this is the experimental timeline for our International Space Station experiment, where we have media changes that occurs immediately upon docking at the International Space Station, and then once a week after that. And throughout this experiment, we have termination of a subset of our samples for fixation in paraformaldehyde, or storing an RNA later for RNA-seq analysis, so that we can get a better idea of what's going on at the molecular and structural level. So these engineered heart tissues were transported to the Kennedy Space Center. Transporting these tissues is, as you can imagine, a very scary process. We have to hand carry them through the TSA and onboard the flight. And we were lucky and successful in transporting our tissues without having any damage occur to them down to Kennedy Space Center, where they were launched on board SpaceX CRS-20. After being integrated into the Merlin incubation system. So after that takeoff, they were successfully boarded onto the International Space Station. This is astronaut Jessica Mayer performing a media swap in this little video clip here, as well as just working on the engineered heart tissues. And as I mentioned, we use syringe-based media changes using those ports on the side of the chambers. An analysis of the flight data showed that we had functional defects in these engineered heart tissues, where we saw significantly decreased twitch forces over the duration of their stay on the International Space Station, with twitch forces staying low after 9 days of being back on Earth. Metrics for arrhythmia also showed a peak at day 19, but a reduction by day 26 and a recovery to baseline upon return to 1G. But we were excited to find these functional deficits and changes due to their stay on the International Space Station and in microgravity. And inside the cell, we looked a little bit closer about what might be causing these changes. And we found very immediately in our late spaceflight samples, large intracellular lipid accumulations. You can see all of these lipids throughout these, and they're very prevalent throughout these late spaceflight tissues, and they're not found in our control or early spaceflight samples. And that's indicative of oxidative stress and or mitochondrial dysfunction. Essentially, the reasoning behind that is that these lipid accumulations would either need to be in the media, and the astronaut Jessica Mayer promised that she did not add any extra lipids into these samples compared to our controls. So as long as we trust her, it has to be due to decreased metabolic activity and inability to metabolize these lipids. And it is also, lipid disorders are also linked to mitochondrial dysregulation, and there's a link between the lipid disorders, mitochondrial dysregulation, and poor cardiac health. So taking just a closer look at the mitochondria, we do see signs of mitochondrial ultrastructural deficits and defects. We see autophagosomes frequently in our late spaceflight samples. We also see linearized cristae in our mitochondria, as well as compartmentalization in our mitochondria, which are all signs of mitochondria damage. These mitochondria are also much smaller than the mitochondria we find in our control samples. All of this is currently being further analyzed and quantified for publication, but we were excited to find these lipid accumulations and signs of mitochondrial dysfunction. So moving to the next phase of the project, while we're finishing up this analysis, we also need to be getting prepared for next October, when we're going to be launching to the International Space Station for our second mission, where we're going to be trying to determine whether we can prevent these negative effects associated with spaceflight. So to do that, we are using a random positioning machine, which simulates microgravity by rotating samples in a random pattern, effectively summing the total gravitational forces felt by these tissues to the levels found on the International Space Station. So we put the tissues onto this random positioning machine, and this is some preliminary data. These experiments are still ongoing, but we found decreased twitch forces. So in our control, we tend to see, we put them on after 2 weeks, and we tend to see increased twitch forces up to about 1 month. But on the random positioning machine, it initially increases and then decreases in function. Similarly, we saw no net change in our control tissues in the mean change in beat interval, but we see a statistically not significant increase in the mean change in beat interval, which is a metric of arrhythmia. But these samples are still in the random positioning machine. We hope to see similar effects of this simulated microgravity to what we found on the International Space Station. So moving to the methods we want to use to prevent the effects of spaceflight on engineered heart tissues. One of the methods we are testing is active and passive mechanical actuation, where here we have a magnet, and with our magnets embedded into the PDMS posts, moving this magnet closer and further away can cause preload on these engineered heart tissues. And similarly, this passive method here, we just increase, or basically we have these bracers that decrease the effective height of the post. These are very stiff and are effectively immovable by the tissues. So by doing this, we decrease the effective height, which increases the resistance to contraction on these tissues, which is similar to an increase in afterload. And we're hoping that this can increase the strength of the tissues and prevent any damage to the engineered heart tissues, similar to how an astronaut exercise regimen would decrease the effect of microgravity on skeletal muscle atrophy. Next, we also plan to test a panel of drugs for efficacy in preventing microgravity-induced cardiac dysfunction. However, as many people have mentioned, PDMS, which is very highly used in the field of microphysiological systems for fabrication purposes, is well known to absorb drug. Two methods by which drug absorption may occur that we've focused on is through hydrophobic interactions or through random diffusion. So to allow for more accurate drug screens, we first decided to determine methods capable of preventing drug absorption. So to address the first problem, which is hydrophobic interactions, we've utilized a PDMS PEG copolymer, where we have our PDMS monomer here and our PEG monomer copolymerized onto the side. And this PEG is a more hydrophilic material, and it should decrease the hydrophobicity of the material. However, don't take my word for it. Let's take a look at the data. So we quantified the water contact angle as we increased the concentration of the PDMS PEG additive. And we found significantly decreased water contact angle, which is used as a metric of hydrophobicity. So we are decreasing the hydrophobicity by including this PDMS PEG. So next we tested whether this PDMS PEG block copolymer additive could decrease drug compound absorption using 5 of the comprehensive in vitro proarrhythmia assay, or CIPA compounds. We tested drug absorption by incubating PDMS samples with drug for 1 week and measuring the drug concentration compared to our standard via UV-Vis. While effective for most drugs, we felt that it could be further improved. We still had pretty high levels in some of the drugs, as well as no net change in drug absorption in Bepredil. So the two causes that we mentioned earlier for drug absorption, the other one, in addition to hydrophobic interactions, is the net diffusion. So without being able to change the laws of physics, we needed to cheat a little bit in order to prevent diffusion into our material. And to cheat, obviously, we just changed the concentration gradient. So we pre-treated for 24 hours the PDMS samples in drug, and then moved them into new drug and tested absorption following 1 week. And this is a lot of data to try to take in a very short period of time. So I have a summary slide on the next one, but a net takeaway we can go through here is just that while effective in some block copolymer concentrations, but not others, it's also effective at some doses, but not others. It's very variable. But what this allowed us to do when we combined both the block copolymer as well as the drug pretreatment is to identify a absorption preventing condition, or APC. So this is a heat map that goes over just all of the experimental conditions that we had, and we identified which condition prevented drug the most. And using this, we were able to decrease the mean absorption to 0.36%, which is very low comparatively, which is also, mean decrease of 92%. So next, we just wanted to make sure that this would enable more accurate drug screens. So we used our engineered heart tissue platform, and tested drug kinetics and the change in twitch forces using verapamil. And this is our positive control where we have 100 micromolar of verapamil. This is the absorption preventing condition, which should nominally have about 100 micromolar of verapamil as well. And then this is the one that has PDMS absorption, which again, all of these should be about the same as the positive control. So any changes are due to the drug loss due to PDMS absorption. And what we found is a closer match with our APC condition to the 100 micromolar positive control. And when we calculate the IC50, we see a statistically significant difference between our positive control and the PDMS condition, as well as a statistically significant difference between the PDMS absorbed condition and our absorption preventing condition, while there's no significant difference between the 100 micromolar positive control and our absorption preventing condition. So using this, we're going to continue to do drug screens and further test these and hopefully come up with some interesting therapeutics to test in October of next year on our next spaceflight mission. And with that, I'd like to thank funding sources and all of our collaborators. I'm sorry I went a couple minutes over there, but yeah, happy to take any questions. It looks like there's a few in the chat.
Yeah, we have time for one quick question and the rest please answer in the chat. So the question from Benoit Cox, are the Earth controls of the cardiac cultures also treated with similar G-forces like during liftoff?
So we debated methods that we could potentially try to do that. Overall, the G-forces experienced during liftoff very low compared to centrifugal forces that are commonly experienced during standard cell culture. So we're not too concerned about the potential for these relatively low G-forces that are experienced during takeoff. And we weren't able to come up with a viable method of causing these high G-forces in our tissues prior to the controls. However, our random positioning machine now is also capable of hypergravity, so we'd be able to better simulate that with the new equipment that we have.
Thank you. With that, we will move on with the next speaker, and please answer the questions in the chat in Q&A.
Okay, thank you so much, Devin and Lina. Our next speaker for this session is Zhang Alan Lee from the University of Pittsburgh, who is also one of the 4 nominees for the Young Investigator Award. Welcome, Alan, the stage is yours.
Thank you for the kind introduction. Good morning, good afternoon, or good evening. My name is Alan, and I'm from the University of Pittsburgh School of Medicine. First of all, I would like to thank the organizers for this wonderful opportunity to present our research. My presentation today is about a synovial joint on the chip system. Joint diseases such as osteoarthritis or OA are highly prevalent and represent a leading cause of physical disability worldwide. These diseases affect all joint components and are often characterized by inflammation and cartilage degeneration. Unfortunately, currently we do not have any disease-modifying medications available for OA treatment or for targeting other joint diseases like joint inflammation. We believe that this huge unmet medical need is primarily caused by the absence of an appropriate model that can enable us to effectively study disease mechanisms and test potential disease-modifying medications. Therefore, the goal of our study is to develop an effective model for replicating joint pathophysiology. And we believe that to develop such an effective model, we need to use cells from human patients, because this can enable us to study patient-specific modeling and drug testing. It uses 3D cell culture to mimic the in vivo microenvironment, and it contains multiple joint components that can have real-time bidirectional crosstalk. We hypothesized that such a human cell-derived multi-tissue microphysiological system named microjoint can model inflammatory and degenerative features of joint diseases and be used for testing potential drugs. To do this, we first used human stem cells and a hydrogel scaffold to engineer 3 key tissue components of the knee joint. Once we have the engineered tissues, we put them in a 3D printed bioreactor to form this microjoint chip. One key feature of our microjoint model is that tissue-specific media, including adipogenic media, fibrogenic media, and osteogenic media, which are indicated by AM, FM, and OM here in this figure, they were separated at the top to maintain the individual tissue phenotypes. At the same time, we also have this shared medium at the bottom of the chip to allow the tissues to have crosstalk and communication. One thing that we have to confirm here is that all the tissue components will be able to maintain the individual phenotypes for at least 1 month so that we can confirm the physiological relevance of our model. From the PCR data here, we can see that the bone and cartilage markers were only highly expressed in the bone and cartilage tissues respectively, but not in other tissue components. Using immunostaining and histological staining, we can see that the bone and cartilage tissues also have their respective tissue-specific extracellular matrix as well as marker proteins. Same here, we can see the lipid droplets being well retained in the adipose tissue and the fibroblast markers well expressed in the fibrous tissue after 4 weeks of culture in the microjoint. Therefore, the results here confirm that we have a model that allows the maintenance of tissue phenotypes for at least 4 weeks. Once we have confirmed that the tissues can happily live together, we then moved on to disease modeling. To do this, we used the pro-inflammatory cytokine interleukin-1 beta or IL-1 beta to challenge the fibrous tissue or FT. To do this, we added IL-1 beta to the FM, but not in other streams, so that other tissues were not directly challenged. After 7 days, we used RNA sequencing, histological staining, and immunostaining, as well as Luminex assay to assess the responses by the FT tissue as well as other tissue components. First of all, when we look at the transcriptome of all the tissue components for the control group and the disease model, we can see that very obvious changes indicating pathological changes in the FT tissue. And we also saw pathological changes in gene expression by other tissue components, including bone, cartilage, and adipose tissue. We then wanted to explore the disease markers. Because cartilage tissue is the key tissue that we want to focus on. So we analyzed the transcriptome carefully for cartilage tissue, and we identified more than 500 differentially expressed genes in the OCC, namely the cartilage tissue of our microjoint model. We performed pathway enrichment analysis, and we compared our model with 2 human studies as well as 1 animal study. We were very delighted to find that actually our microjoint model shared more commonly enriched pathways with the human patients than the animal model did. And when we look at the common pathways between the human study and our microjoint model, we found that many of the common pathways are highly relevant to osteoarthritis. We then moved on to Luminex assay to identify disease markers by assessing the components of the simulated synovial fluid. Earlier, I mentioned that this is a shared medium by all the tissue components. Therefore, this can give us a very useful data to show the health states of all the tissue components. Here we can see that compared to the healthy control, the disease model had very significant increase in the concentrations of many inflammatory and degenerative markers, including IL-6, IL-8, and also PGE2 and MMP13. When we look at the fibrous tissue, we found that the fibroblasts had very high levels of apoptosis due to the inflammation and also high expression level of MMP13. Because of tissue crosstalk, we found very obvious degeneration of the cartilage tissue in the histological and immunostaining images shown here. Therefore, we have confirmed that the inflammation in the FT was able to induce inflammation and degeneration of the cartilage tissue And this was a successful establishment of joint inflammation and degeneration model. To explore the utility of the microjoint model in drug testing, we used the two different methods of drug administration. The first one is called systemic administration, and we used a very common NSAID that has been prescribed to OA patients in many clinics. Here we added naproxen to all the medium streams shown here, as indicated by the black arrows, to mimic the systemic administration of the drug. In the RNA sequence data, we can see that the changes in all the tissues indicated alleviated inflammation and less degeneration. And similarly, we performed Luminex assay in the simulated synovial fluid, and we found that the decrease in the concentrations of many degenerative and inflammatory markers, indicating the protective effect of naproxen. We also used histological and immunostaining for the fibrous tissue as well as the cartilage tissues. And we found that, again, similar trends were seen for the effect of naproxen on the alleviation of inflammation. Next, we tested a second mode of drug administration, which is intra-articular administration, using the 4 drugs here listed on the left. We added those drugs to the simulated synovial fluid only to mimic intra-articular administration in clinical trials. From the gene expression data here, we can see that the chondrogenic markers could be upregulated upon drug treatment. At the same time, the inflammatory and degenerative markers could be downregulated after 1 week of intra-articular administration of 4 of those drugs. Again, in the Luminex assay, we can see that the concentrations of many degenerative and inflammatory markers could be decreased after the drug treatment. Taken together, the results from the systemic and intra-articular administration of the drugs can show that the disease model established using our microjoint could be potentially very effective in predicting the in vivo efficacy of potential disease-modifying drugs that target joint diseases such as OA. In conclusion, in this study, we have established a 3-dimensional human cell-derived multi-tissue chip that can be used to model the inflammatory and the degenerative features of osteoarthritis. And we also demonstrated the utility of the model in potential disease mechanism study as well as drug testing. Moving on, we are now incorporating macrophages in bone, synovium, and adipose tissues to provide our system with an immune response. And we are also introducing mechanical stimulation capability to study how the mechanical forces affect the health of various tissues, especially the cartilage. And here, this image shows a prototype of a mechanical stimulation device that we are currently developing to integrate with our microjoint model. Before I close, I'd like to acknowledge all the support from my mentors, as well as our collaborators from Pittsburgh, Stanford, UNT, and Maryland. And I'd like to acknowledge the funding support from NIH. Thank you, and I'd like to take any questions you may have.
Thank you, Alan. I'm sorry you had to deal with the Zoom ghost. I have a question about macrophages. So how are you going, can you give us some insight? What is your ideas, how you will incorporate that, and what it will be this iPS derived, so how you will generate them?
Thank you for this very important question. Regarding the incorporation of macrophages, we are in close collaboration with Dr. Stuart Goodman's lab in Stanford. So the idea is to incorporate M0 macrophages in bone, adipose tissue, as well as synovium. And we want to see how the inflammation, for example, in the fibrous tissue would affect the phenotype of the M0 macrophages in FT as well as in bone and adipose tissue. Actually, in our preliminary results, we have already seen very huge effect of the macrophage phenotype on, for example, bone mineralization and also in the adipose secretome. So I think this is a really exciting direction that we are heading to, and I look forward to more data from the macrophage incorporated microjoint model.
And what would be your cell source for macrophages?
Previously, we have been using periphery blood-derived monocytes, and then we use MCSF to differentiate those monocytes into macrophages.
Okay. Are you planning to work on a patient-derived systems cells to generate your models toward personalized medicine?
Right. This is a very important question. Currently, we are only using mixed cells because we need to use a really large quantity of cells. We in my previous experiments, we used cells pulled from 20 patients. However, with the development of iPS technology, we are already developing several cell lines of iPS cells from different patients, and I don't see any problem in developing patient-specific, personalized microjoint model. And our recent publication on the osteochondral tissue was actually based on iPS cell. Currently, we are doing other tissue components using iPS as well. Once we figure out the optimum condition for deriving those tissue components, then they would be integrated with the osteochondral tissue in the microjoint, and those microjoint chips would be fully personalized.
Great, thank you. And we don't have at the moment any further questions, so Ricardo, we can move to the next speaker. Thank you, Alan.
Thank you.
All right. Thank you so much, Alan and Dina. And now I would like to call on this virtual stage our next speaker, Michelle Brouwer from Mimetas. Welcome, Michelle, and thank you again for your flexibility.
Yeah, so good morning, everyone, and good evening for everybody in Europe. I'm Michelle Brouwer, and today I'll tell you a little bit about about T-cell and vasculature interaction in the organoplate graft, which is developed by the company Mimetas that I work for. So at Mimetas, we focus on developing human tissue models for better therapies by unlocking novel biology, creating better and faster assays, and to have an impact on therapy development. And the platform we work with is the organoplate graft, which I show you in the next slide. But you can use these to create more advanced tissue models using 3D cell culture. You can also look at creating tubules, so either vessels or epithelial tubules, so for example, gut tissue. And it's very suitable for studying cell-cell interactions, looking at cell migration, but also invasion, immune cells and tumor cells, of course. You can also do angiogenesis assays and look at vascularization, for example, of spheroids, as you see here in the picture, but also, for example, explants. So this is the organoplate graft. I'll just play you a short video. This is one of the plates that we have. So this is the organoplate 3-lane. And basically, different plates have different compartments, which are separated by something called a phase guide. And this is not a membrane, but really only like a small sort of barrier against which we can create different hydrogels and culture different vessels and different tissue. And as you can see, you can load different compartments in it. So for example, here on in the red space, you can culture vasculature, and then in the middle you could have like a fibroblast with immune cells, and then on the other side you could have a gut epithelial layer. And as you just saw here, is and we also place our plates on a rocker, which creates this perfusion throughout the compartments, allowing cells to move through it, but also fluid to move through it, and also for the model to develop in a more physiologically relevant setting. So the importance of 3D models is really studying the interaction between different cells. So in this example, it's lymphatic endothelial cells, which are grown in the top plane, and these are colorectal organoids. So really looking at the interaction between these different cell types and how they influence each other. And this is from the paper by Frankel et al. Published earlier this year. And we have different plate formats for this. So you just saw an example of the 3-lane 40. We have a similar plate called the 3-lane 64, only this doesn't have an outlet in the middle compartment. Then you have a 2-lane if you just want, for example, 2 different tissue layers. And we have the organoplate graft, which allows for slightly bigger tissue culture. So, for example, think of spheroids, but also explants, or a lot of different tissue cells. And one of the things I want to show you today is the interaction between immune cells and vasculature. And in this case, you see T cells that are adhering and transmigrating through a vessel, and they are shown in green. So these are CD3 T cells. And as you can see, you can nicely study how they migrate out of this tubule. And you can enhance this using different techniques. So one way is, for example, by treating the vessel beforehand using TNF-alpha. That way you upregulate the ICAM1 expression, and you also see enhanced T cell attachments afterwards, even after you wash the TNF-alpha away. And you can really nicely monitor the immune cells in these plates. So you can really look at how they behave. So this is just an example of an endothelial vessel with some T cells added to it. So there's nothing in the bottom compartment that would attract them to go there, but you can really follow how they move around in the plate and in the vessel. And you can then, so here you, for example, have another vessel. And in this case, we have hydrogel in the central compartment, collagen gel. And then in the bottom compartment, we added a chemoattractant. And you see how these T cells nicely migrate from top to bottom. You can really nicely quantify this attachment and migration. These are some figures from the organoplate graft. So here there is one piece of vasculature, and the other one you have another vessel, and then we add something we call a sprouting mix to get these vessels to form new sprouts. And then in the middle there's a spheroid, but this one isn't stained in this figure, so you can't see it. But we added immune cells on both sides, and then added the chemoattractant in the center to attract these cells to the center of the compartment towards the spheroid. And you can really see that these cells go into these sprouts and go towards the central compartment. And like I mentioned, you can quantify this really nicely. So when you add, for example, different TNF-alpha concentrations, you see as you increase this concentration, the amount of attached cells increases. But if you also stimulate these T cells beforehand, you see this increase even further. And something else you can do, as I mentioned just now, is you can also add chemoattractants. and then you see a similar effect. And here we also looked at CD8 T cells where we combined both stimulation and chemoattractant trigger. And here you see, especially in the stimulated cells combined with these triggers, you see a huge amount of cells migrating to the graft chamber center. So you can also do this for disease models. And on the left, you see an example of a tumor compartment where there was a vessel in the top, and then we added immune cells to the vessel, and these immune cells migrated out of the vessel, and they migrate to the bottom compartment where we have tumor cells located. And then we had a condition where we added unstimulated T cells, a condition where we added stimulated T cells, and where we primed the T cells actually with the tumor cells itself. And then our quantification tool allows us to really look at the distance they've migrated. So in these dotted lines, you see basically the interface. So you see here are the cells that remain in the vessel. And then here you see the cells that are present in the ECM at the moment of quantification. And then here you see the cells that migrated really towards the tumor. So you see that if you stimulate or expose these T cells beforehand to these tumor cells, they recognize it and they migrate to the bottom compartment. And of course, you can also nicely show this in a histogram. On the right, I have another example where we looked at T cell motility in response to antibodies. So we cultured a squamous cell carcinoma cell line, and we either added unstimulated T cells, stimulated T cells, or T cells that were already exposed to these tumor cells beforehand. And then we added two different types of treatments. So treatment 1 and treatment 2. And when you look at these results, the biggest difference you see is when you combine these two treatments. So when you have both treatment 1 and treatment 2, you see in all the conditions way more cells migrating. And next to migration, something else that's of course, especially in immune oncology, a big topic is immune cell function, because I mean, these immune cells can migrate, but are they really functionally properly, or are they being suppressed? So one functional readout, for example, that you can have is T cell killing. So here on the right, we stained both the T cells in red, and we have a cell death marker in green. And that way you can quantify the cell death over time. So you can follow this in real time in organic graphs. And in the middle, we have another example of priming, adding prime T cells to the tumor, just the T cells, and then the unstimulated and stimulated conditions. So I've shown in this presentation that we can look at really closely at this interaction between T cell and vasculature co-culture in a high throughput microfluidic platform. Something I maybe haven't mentioned yet is, so this is a plate where you can have 64 different chips per plate, so 64 different conditions basically. So this family is very suitable for the development of advanced immune models as well as readouts. Something I haven't shown in this presentation, but it's definitely possible. So you can also isolate the cells out of the plate, and you can phenotype them. Depending on their interplay, they will probably have a very different phenotype. And you can study this interplay between immune cells and their environment really closely. So we offer high resolution information on T cell attachment and transmigration, possibly under the influence of cytokines, but also under influence of treatment. And we have the ability to study T cell recruitment towards the chemoattractant tumor or inflamed tissue. Yeah, so these are some of the different formats you can choose from. And there are a lot of different analysis we can do. So I just mentioned, or also flow cytometry, but also RNA-seq. We can look at the proliferation of these cells, the viability, and also adding other immune cell or tissue compartments. And then I would like to thank you all and ask if there are any questions.
Thank you, Michelle. So there are a couple of questions. The first one is from Andres Denies. How long endothelial cells function in the system? Are they changing during this period?
Yeah, so overall, of course, when we start the cell culture, they're still growing, so they're forming a tubule. But after that, they're quite stable for a period of time. So we have cultures that can be grown for about 14 days.
Next question is following. In order to assess T cell migration, is it better the 3-lane format or 2-lane format?
I think it depends really on what you would like to model. So the 3-lane really offers you an extra compartment. So for example, what it could be interesting for is if you want to look at the role of stroma in immuno-oncology. So really including also fibroblasts and maybe also other compartments in between. But if you just want to look at how these cells migrate directly out of the vessel and into, for example, the tumor compartment or another tissue, then I think the 2-lane is more suitable and easier to use.
Thank you. Next question is from Silvia Scaglioni. How many immune cells do you see and which is the percentage of their infiltration?
Yeah, so it depends a little bit on what you want to look at, but for example, about 10 to 40,000 immune cells. And we see, like in really good conditions, about 25% goes in, and depending on the other microenvironmental cues, it goes down to 10%. But if it comes below 10%, then I think it becomes lesser.
Okay. The next question is from Divya. Michelle, how does the percentage of tumor cells cells in one of the compartments affect T cell migration?
Yes, so that is really tumor dependent, and we see that in a lot of different models, it really differs. So some tumors really attract the immune cells from themselves, and otherwise you might need to add chemoattractants to really stimulate them to migrate towards the tumor. So some tumors really seem to block the immune migration, while others really sort of stimulate it, and then basically upon arrival of the immune cells, they change the immune cell phenotype. So that's actually really interesting to see.
Mm-hmm. And the last question for now I have for you is from Katarina Apostolopoulou. How can you isolate the T cells from the plate after the migration experiment? Can you do it from all the channels?
The answer is yes, but I can't share the technique openly. Then please contact me through my email address and we can get in touch.
Okay, that's it all for now. Thank you again for your presentation, and we are ready for the next talk.
Yeah, thank you very much.
Okay, so thank you so much, Michelle and Lina. Our next speaker for this session is Colette Bichel from Roche. Welcome, Colette. The floor is yours.
Hello, thank you very much for giving me the opportunity to present. So I'm excited to talk to you about the work that we're doing here at Roche in pharmaceutical science and with the ITB. So I will be talking about our blood-brain barrier models that we use to look at improving drug delivery to the brain, and also recent developments that we've done on these models to include cells from the brain parenchyma. Yeah. So as you all know, probably it's very difficult or challenging task to get drugs delivered across the blood-brain barrier. And this is due to the structure and the function of the cells that we have at the blood-brain barrier. So the brain endothelial cells really have specialized junctions that are very tight. They have a specific set of influx and efflux transporters, and there is also pretty low vesicular transport compared to endothelial cells from other organs. And together with the pericytes and the astrocytic end feeds in the basement membrane, they build this neurovascular unit that is very tightly separating the blood space from the brain parenchyma. And so to improve drug delivery to the brain, we can actually harness the endogenous brain-specific transport receptors. And here I would like to show an example that we use at Roche for this brain delivery platform. So this on the left, you see an animation of the brain shuttle that is targeting a transferrin receptor, which is highly upregulated in brain endothelial cells and less expressed in other endothelial cell types throughout the body. So if we couple this brain shuttle construct to a therapeutic antibody, we have this platform that is really improving drug delivery to the brain. And so upon binding of the brain shuttle with the cargo, there is uptake and transcytosis of the whole cargo across the blood-brain barrier. So we are working a lot, as I said, on these constructs. And the question is how we can accelerate the development and optimization of such platforms. So we believe that human in vitro models are really good to do that, because we can investigate brain specificity and also have really human cell types to evaluate transport efficiency. We can assess human-specific receptor expression, and then investigate the biodistribution if we also include other cell types behind the blood-brain barrier. And we can go towards disease modeling using human iPSCs. I will touch on that a bit later as well. First, I would like to introduce you to the BBB organoid formation assay that we have developed in-house. And this has just recently been published by Claire Simoneau. We use the Sun Bioscience platform, which is basically based on a 96 well plate format. And in each well, we have 55 microwells where the spheroids or organoids can be formed. So this allows us to really generate a lot of organoids per plate, about 3,300 per 96 well plate. We use human brain endothelial cells, pericytes, and astrocytes, and actually they self-assemble to form a kind of a blood-brain barrier structures in vitro. So the endothelial cells line the outside of the spheroid, and then we have astrocytes that build or aggregate in the core, and the pericytes are also part of the core, but they're more close in contact with the endothelial cells. So we wanted to see if these BBB organoids can reproduce some of the blood-brain barrier properties that we want to test. And so for this, we exposed the organoids to dextrons. And indeed, if we have an integrated full BBB organoid, we see almost no penetration of fluorescently labeled dextron into the spheroid core. But when we don't have the endothelial cells, we do see signal in the core, and we can quantify this. And so we saw that compared to untreated spheroids, the BBB spheroids that are treated with 4 kilodalton, 40 or 70 kilodalton dextrons are actually also impermeable to all these sizes of dextron. So we have a tight barrier. And then after having established this, we used the system to screen and look at transcytosis of the brain shuttle constructs that we are using in-house or testing in-house. On the left, you see an example of this. So using the transferrin receptor targeting human brain shuttle construct that is fluorescently labeled, we see that it can accumulate in the core over time. because there's active transcytosis. And this is not the case when we use brain shuttle that is targeting the mouse receptor or a non-targeting IgG, and we can quantify this here on the right. We then use this in a screening assay to compare different variants of the brain shuttle construct and compared it to the original construct to see if we have increased transcytosis or less transcytosis. And we can now use this to really screen for good variants. What is also interesting with this platform is that we can use the BBB organoids to look at the mechanisms. And here I show you an example where we did a CRISPR knockout in endothelial cells. And then we looked at the function of the transcytosis, basically. So if we knock down the transferrin receptor, as well as clathrin, which is also involved in the process of transcytosis, we see much less uptake of the brain shuttle construct. And this is not the case when, for example, we knock out caveolin-1. So in the future, we can use this also to screen to find more mechanistic aspects and to screen for maybe novel receptors that are important in transcytosis that it could potentially target. And then to go a step further now, we're also interested in looking not only at transcytosis, but also at what happens after the construct crosses the blood-brain barrier. And for this, we are looking into the next generation BBB models, basically where we want to integrate brain parenchymal cells. And we would like to do that because, as I just mentioned, we would like to assess also how the drug is distributed in the tissue afterwards and look at the function of target cells in the brain parenchyma. We are also interested in investigating how the drug pharmacology then impacts the barrier function, whether there is a back and forth, for example, in barrier permeability. And we want to use it to explore barrier functions in disease models. And these are the proposed models that we are working on. So I already mentioned the spheroid organoid model that we are working in-house. So we are looking into integrating microglial cells and as well as neurons into this platform to have a more functionality and also develop functional assays. The advantage of this system is really to be able to do screening, because we can, as you saw, we can have a lot of spheroids on one plate. And then we are also looking into a little bit less high throughput screening method, but that model that would be useful to also investigate the mechanisms or have a better view on the different cell types, which is a model of perfusible vessels. And interestingly, in the previous talks, this model or similar models have also been mentioned. So I think it's very interesting also to explore these different models. So first, I would like to talk to you about embedding neurons and microglia into the BBB organoid arrays. So to first test if it's even possible and to figure out the best medium composition such that all the cell types are healthy and functional. We used Lumes neurons that we can differentiate within a week or 10 days, and we were able to integrate them into these organoids. You can visualize them here by the Touche-1 staining. And we also used iPSC-derived microglia to integrate into the organoids. We have both in-house iPSC-derived microglia, and we also purchased some from Fujifilm. And again, we used the Sun Bioscience platform to do these assays. I also want to mention to you the neurovascular unit model that we work with. So we use the chip from A&M Biotech with a central channel for vessel formation, and we also use an in-house adapted chip. And then we seed here primary human brain endothelial cells, pericytes and astrocytes, and look at the vessel formation for 7 days. So we see that we do have perfusible, accessible vessels. And interestingly, the pericytes line really closely the vasculature, and we think that it stabilizes them. And there is also contact with the astrocytes in the chip, as you see in the animation. So we're working currently on integrating the other cell types also in this model, namely the microglia and the neurons. And we also would like to go more towards using iPSC-derived cells to, in the future, work with patient models. So I'm very excited to share with you some first results that we got when integrating iPSC-derived NGN2 overexpressing neurons in this neurovascular unit platform. So the nice thing about this system is that the vessel formation takes place over the first few days, and we have mature vessels within about a week. And this is the same time span that is needed for the NGN2 overexpressing neurons to really form neurites and develop as well. So we can do this process in the chip in parallel. And then we are also working to ameliorate the protocol to get iPSC-derived endothelial cells that have a real brain and microvascular identity. And we're excited also to in the future integrate them in the chip as well. And the first goal, once we will put together this system, will be really to look at the question of how does neuronal cell death and impact activity and the neurovascular integrity. And we think that we can really start to answer this question when we have all the cell types in place, namely the endothelial cells that form the barrier together with the pericytes and astrocytes, when we also have the neurons that we can then induce cell death, and as well as microglia that are responsible for the large part of phagocytosis. And then we can go towards disease modeling. So this was a short overview of the activities that we're doing. And to summarize, we really achieved to have a reproducible and high throughput 3D in vitro platform to look at transport across the blood-brain barrier, to look really at transcytosis. And this we do in this spheroid organoid model. Furthermore, we also managed to get self-assembled vascularized blood-brain barrier CNS chips. So we integrate and mature neurons and also microglia in this chip, and we can in the future use them for functional assays. So our vision is really to go towards an integrated in vitro system where we can use one system for a holistic evaluation of the blood-brain barrier function, but then also of drug targeting and biodistribution behind the blood-brain barrier, in the future doing PK, PD, and tox studies all in one system. We really think that by also looking at different platforms, depending on the needs, that this could be useful. For example, we see that the spheroid and organoid platform is really amenable for high throughput screening, and to see to look for also new regulators of transcytosis using these mechanistic assays. And then we're also interested in discovering new pathways and figure out cell-to-cell interactions and regulation at the neurovascular unit. And this will be done in a bit less high throughput system, but more amenable to deep investigation, such as this perfusible chip system. And with this, I'm already at the end of my talk, and I would like to thank you a lot for your attention, and I'm happy to take questions. I would also like to first thank all my team members in PS and NRD in the different units at Roche. It's really a pleasure to work with everyone, and also I would like to thank our external partners, and I'm happy to take questions.
Thank you, Colette. It's a really interesting talk, and I have tons of questions myself. But first, I will go to the Q&A now. The first question is about how big is your organoids?
How big they are in terms of size?
Yes.
So they are, I think, about 300 micrometers in diameter, something like that. Yeah.
Okay. And then I have two questions from Winfried. One of those is also about the size. The first question is, it's very great talk. Do you consider to optimize, for example, the size of the blood-brain BBB spheroids, cytinoids, the geometry, size, cell number, composition, etc., influence the function properties of the BBB spheroids? And there's a link to the paper you can look later. But so do you work on optimization of these parameters?
Yeah, it's actually, it's a constant optimization, right? Yeah, especially when integrating new cell types. I think we're not only concerned about the size of the spheroid, although, yeah, this is also a consideration, but Also, we're concerned about the nutrient accessibility in general. Well, the size obviously has an influence on that, but also the medium composition itself, that we can really have a cell culture medium that works for all the cell types and doesn't, for example, activate one cell type too much and then influences, for example, the integrity of the blood-brain barrier. So we're carefully evaluating the medium composition when it comes to integrating new cells.
Great, thank you. And follow-up question from Winfred, where also I had a similar question again about the size and and how long can you keep them in those plates? Because obviously without perfusion, we can deal with hypoxia, core, and similar effects.
Yeah, so I mean, we do check for cell death with a caspase 3/7 staining usually, and we don't see an issue with that. So we don't have them in culture for a very long time before we do our assay. We really, what we want to do is put together all the cell types and have them rearranged to have this BBB structure. And once we have that, we are ready to do our assay, and this is achieved within 2 to 3 days usually. So we're not reliant on very long-term culture. And yeah, as I said, we generally test for apoptosis, and we don't see excessive cell death, for example, in the core. So we're not concerned with the hypoxic core.
Okay, and the last comment from Winfred, it's real congratulations to your great achievements with the BBB spheroids. So and the next question from Andras Dimias, great progress. How to achieve a long-term matured neural network with all cell types remaining viable and functioning together?
Yeah, that's a good question that we also try to answer. Yeah, so I think for longer term studies, we would go for the perfusible vasculature model, because we've noticed that once the blood vessels are perfusible, they're actually quite stable, even over weeks in culture. So the integration of the neurons, for example, these NGN2 overexpressing neurons, is relatively new, so we don't have very long-term results yet, but we can have them in culture and happy and alive basically for 1 week, a little bit more. 8 days, I think, is the maximum for now, but I think we could probably go further. Yeah.
Thank you. I have a couple more questions. So how is it done technically that the endothelial cells wrapping up so tightly and form all those tight junctions? Is it just the biology behind that, or you have some tricks you do technically?
I think, no, I mean, it's not magic involved. I think it's the cells themselves, the endothelial cells tend to, how do you say, like, cover an extracellular matrix and then have polarize themselves basically to have one luminal and one unluminal side. So it kind of makes sense for them to align on the spheroid surface. We did observe some differences with like using different cell lines or primary cells. So not all cells do the nice tight layers, but we basically based on our work on a publication from 2013 that I forgot now the name, but I can later on like write it in the chat, where it was first described, this model was first described.
Great. And another question I have on the microglia. So when you combine that with the luminous culture, so how long then, first of all, what stage the microglia is at? Is it already mature microglia, or is the microphage progenitors? And when you incorporate that, how long you can keep those spheroids in culture?
Yeah, so we have not optimized yet the co-culture of neurons plus microglia, because that's really tricky to have, especially in a long-term culture. Regarding the state of the microglia, so the ones that we get in-house are basically derived from premax that have been then pushed towards the microglia for 10 days. So they're relatively mature, and we integrate them as is, and then basically use them in the assay 2 days later. And then the one that we purchased from Fujifilm, they're also already differentiated, and we purchase them like this. They're characterized in-house at Fujifilm.
And how you optimize the medium for that? Do you change the neuronal medium to add more growth factors for microglia, or the cells provide?
Yeah. So ideally, it would be great if the cells provide some factors to each other. But for now, yeah, we're still in the phase of testing. So the main idea is to basically to have like a medium that we use for the spheroid that has also some growth factors for endothelial cells and an astrocyte growth supplement, and then we add neuronal factors to it. And then we also add some factors that we know that are necessary to keep the microglia mature, like IL-34 or MCSF. And we're in the process of evaluating whether this negatively impacts other cell types. So I don't have a definitive answer, but we just try to combine what we need is necessary for each cell type, and then just subtract what we don't need necessarily or what could be harmful.
Thank you. So I left one question unanswered in the chat because it's the link. So I want you to see that and click the link if you want. And with that, we don't have any further questions. And Ricardo, we now move to the voting of our young investigators.
Thank you very much.
Okay, so thank you again for the great talk, Khaled and Lina. I would like to thank all the speakers once again for presenting their terrific work and exciting results. In the next few minutes of our meeting are dedicated to voting for the Young Investigator Award. This slide that I'm sharing with you, and please let me know if you cannot see it. This slide captured the 4 finalists of our abstract competitions. These 4 are therefore the finalists of a competition that saw over 22 abstracts screened and ranked for novelty and scientific relevance to the meeting. I wanted to kind of invite you all to participate in the voting, and we will resume in the next 5 minutes. Well, I think we managed to reach the quorum here, so I'm happy to stop a few minutes in advance this voting session.
Okay, I'm gonna end the poll then. And tell me when you're ready to share the results.
I think so. What we will do now is to help all of you had a chance to vote for the Young Investigator Award, and I will have the pleasure to announce the winner shortly after our panel discussion. So as per our agenda, the next half an hour of the meeting is dedicated to panel discussion around the topic of systems engineering, microphysiological systems. The panelists for this session include Dr. Linda Griffith for the MIT, Alison Maltry, University of California, San Diego, and Peter Laskill from Tübingen University, and Dr. Yu Marks from Tissues, and Dr. Yasuyuki Sakai from University of Tokyo. And now I'm pleased to virtually pass the mic to Adrian Roth from Roche, who will moderate a panel discussion. And Adrian, the floor is yours.
Thank you very much, Ricardo. And yeah, welcome everyone to the panel discussion. We have a great round of diverse researchers from the field. and the organizers of this Congress kindly provided us with a few points to discuss here. And so I would right away start. So we've seen today yet another set of really great examples, use cases, new models, new approaches on the horizons, new ways of how to engineer cells. So we would like to first discuss with, so what is in your view the best way to benchmark those samples, so to really get a bit of a feeling what has chances for success, what is something that will move forward, and are there gold standards, and is it the right time actually to define these in order to support the field, or should we leave it open and let the different approaches flourish? What do you think, Linda?
I think it's incredibly dependent on the question being asked. My lab, we spend probably 2/3 of our time on defining what the in vivo biology is and what needs to be replicated in vitro. I am very involved, for example, in studying diseases of the uterus, and there we just have an unpublished data that I can only share generally from a collaborator in Edinburgh, where our in vitro endometrium, we compared against a whole bunch of different drugs to samples from patients who took those same drugs over a time. So it's a longitudinal study in patients, a longitudinal study in cell culture with multiple different donors. And we actually compared two different tissue inert formats with Matrigel and a synthetic hydrogel that we created for multiple cell type co-culture. And there, looking, we're benchmarking against the actual patients who took the actual drugs. And for me, I want to see that kind of benchmarking, in vitro, in vivo. But other diseases, I think the field is kind of sorting through, and each company or academic investigator decides, for each specific disease case, what are the metrics that need to be the same? So I think we need to do a lot of in vivo, start doing that, but it's got to be for very specific questions and looking at very specific pathways or things like that. It's got to move on beyond, metabolism and toxicity. Got to move on beyond that.
Thank you for that. So we heard very, very, very case dependent, and of course the patient is the ultimate gold standard. So what's your take on this, Alison?
Totally agree. I think the more we have in vivo benchmark or gold standard, so we can push the in vitro model to reach those standards, the better. My lab focus mostly on brain organoids, and the major difficulty we have is what is that gold standard, because we cannot really get health human baby tissue, fetal brain, especially if you're looking at a functional level. For example, we do lots of electrophysiology.
I asked you the question.
Yeah, how would you do that, right? So I get these questions over and over. Sometimes, even reviewers, you have to validate that in vivo. Yeah, well, how can I do that, right? So there are things that we just cannot do it. I'm actually trying to rephrase the question now. I'm using the organoid to infer what happens in the very early stages of human embryology. Because really, except if you go to animal model, which also has some limitations, there is no other way. Some people like to use abortus material. This is hard. We also use them. Sometimes they don't come with good quality. You don't even know what is the embryonic stage or the brain region that you are talking about. So I always look with a grain of salt when I see this kind of validation, but that's the best we can do. So that's when we're trying to mimic human biology. But there are other things that you don't have to mimic human biology. Maybe you're perfectly fine just defining whatever cell types or interaction you have. And again, to answer a specific biological question, what is the interplay between microglia and neurons? If you have a model that can do that, it's a starting point. It doesn't have to reproduce exactly what happens in the brain, but it solves you some molecular basic questions. So that's how I see it.
Okay, thank you very much. Uwe, you are a known pioneer working on highly complex systems where you even connect different types of organs. So what's your take on benchmarking and setting gold standards in order to assess whether such a complex model is actually useful?
Thank you, Adrian, for that question. I would go for your proposal and still not answer fully on all these questions, but leave it open. So we are really, for the last 5 years, involved in that 16 different human organ models from customer sides, from research sides. Pharma and cosmetics are different. The context of use is always different. At the moment, we are using primary human cells from healthy people primarily. So like 70% of our operations, or those of the customers of our systems worldwide, are using healthy primary tissues, either commercially available or from patients, to then build in the disease models. Less primary from patients. And as you know, we are continuously working on iPS-derived models in order to make autologous full mini organisms on a chip. This implies either iPS cells from patients or healthy, again, people, and then moving them into the disease area. So whether it is stem cell based or primary, at the moment it's more primary, but we see a very good progress in making iPS, or like stem cell based tissues, differentiating them in multi-organ chips into fully functional human equivalents too. But we still need time, and I just would leave it open because we have a lot of context of use or fit for purpose assays with certain partners where it's quite clear described, it is qualified, and it works for that fit for purpose. And that is currently primary cell based. Only a few systems are fully iPS based, and we have 2, 3, and 4 organ systems in place.
Okay, thank you very much for your answer. So now I would be interested to hear from you, Yasuyuki-san. You're focusing on the engineering part. Would you say that such potential standards or gold standards to benchmark the systems should also apply to sort of the hardware used? We heard a lot about cell sources, the right readouts, benchmarking to patient outcomes. So what's your take on the hardware?
Yeah, it's a very difficult question. So because I'm an engineer, I want to reply from the engineering point of view. So, but basically, the total performance of the MPS is decided by the combination of the device and the cells. And probably, practically, we may focus on the readout. So I very much appreciate the effort of MPS affiliate of IQ Consortium people, US. And they proposed just for main organs, they proposed a kind of standard functions that should be evaluated. So in addition to that, probably we may, in the future, we may make a kind of chemical standard set to assess the readout and compare the different systems. That is practically a way I can think. Yeah.
Thank you very much. So Peter, you are at the interface with biology and engineering. Do you think there's some aspects around the hardware, the engineering part, where we can already talk about certain standards?
Yeah, I think absolutely. There are a couple of aspects where you can look at standards, and the technology side is maybe the easier standardizable. And I mean, there's a lot going on in the fields of microfluidics, just standardizing where ports should be, how you can interface things. I think this is something we can learn from in the organ on chip field, and it would be smart to take up these standards to be intercompatible. And other things, I think it's important to also be able to standardize the readouts and the way you report things. But on the other hand, you also have to be careful to not over-standardize, because that will hinder innovation in the end. So the more you standardize things, the less you leave space for innovation. So I think the clear balance has to be to figure out what can we easily standardize, what has been standardized in other neighboring fields, and where do we need the flexibility to be able to actually innovate new things.
Thank you very much. So we heard now quite a bit about what I would call has been at the foundation for this whole field. So which is on one hand the bioengineering and on the other hand cell biology. So I would like to hear a bit from the group here. So what is kind of the next important field or stakeholder group that should help, that you would like to team up with, and that will help move the field forward? So to give you an idea, we could talk about making use of real world data or modeling, or is it clinical organizations? We heard about patients as a gold standard. Now, when talking about disease models, would patient advocacy groups, could they be of help? So kind of, I would like to hear a bit from you guys. What are the key partners that help move this field forward? So who would like to speak to that?
I can go if nobody's speaking up. I think a challenge, Adrian, for all of us, I mean, I'll hear from others, is it's often hard to get the funding for the people you need from all the fields that you already know are involved. So to, when you're in academia, to have a program where you've really got good PK people, where you've really got, the cell biologists for your tissues, where you're connected to the clinic. So I think it's partly people are involved from these different fields, but getting them all in one room in one program, so you've got everybody covered, is a challenge, and other people can speak up. It might be easier in a company. The one thing I would say would dramatically change my life right now is, because I work in chronic inflammatory disease and modeling, automation of how we handle all the human tissues, the organoid, automatic organoid culturing system. I mean, we spend so much time culturing intestinal and endometrial organoids to make our tissue banks, because we have to do it for multiple patients. And automation around the preparation of the primary cells we need for our studies would be a game changer. And maybe that's out there. So if anybody knows of automated organoid culturing systems that'll culture your epithelial organoids and passage them and all that kind of stuff, please let me know.
Yeah, so that's for scalability and robustness, and also increasing throughput and having enough samples at hand. Yeah,
yeah, from the clinic, because I've got great clinical connections. It's just that whole getting things from the clinic into the devices is a pain point.
Yeah. So the automation folks should help the field. So what other ideas are around? What's
Maybe
the aspect?
I think obviously any connection to in silico, big data, AI, I think this is the obvious next step. But I think even before moving there, the field has been driven a lot by engineers, biomedical engineers, bioengineers, and biologists. And just the easiest next step to involve pharmacologists and toxicologists really right in the core of the field, and also clinicians, the end users. This is already only a small step, but having people that have experience with that in a team that is developing systems and setting up assays is already absolutely crucial, and brings the entire field forward. So I think, sure, there are automation, the big data, and all of these things that will in the long term be the next levels, but even small steps involving all these different fields can be very beneficial.
I already have those in mind. So I go in the OR and collect, the clinicians and all of those people are involved in my projects. So I think we're all in different states of where our pain points are, might be a way to think about it.
I would like to echo what Linda says about funding. because these are quite expensive technology. And especially if you are in the disease modeling part, requires multiple patients, multiple controls, sometimes genome edited cells to mimic the disease. So these are, I mean, it's a long project and quite expensive. It's almost like doesn't fit anymore on the traditional R01 or the expectation for a grad student to stay 4 years. I mean, projects in the lab are taking like 6, 7, sometimes 10 years to get publication. So how do we do that? I mean, we no longer have like one person per project, it has to be multiple.
Yeah.
And then, I mean, you have to be multiple funding sources, thankfully to NIH, but I think that's a part where foundations should chime in as well, and maybe industry. And I know that sometimes industry get a little bit more reluctant with new technology. And maybe one thing that needs to happen is we should start collecting examples where this organoid technology really becomes translational, right? I mean, what you see in a dish is that what you see in the patient. So the more we have those examples, the more convincing the technology becomes.
Yeah, we're starting to do that with some companies. It's you said exactly. I mean, it's just getting the right team for the right period of time. It's excruciatingly, painfully difficult.
Okay, interesting. Any other thoughts on this?
I can add from our side. So we see these 2 or 3 areas even interconnected: automation, software for automation, and not only automation of the pre-culture, but also automation of the complex chip culture and assay performance. And there you need really already software and tools, imaging tools or other automated tools, where our software specialists are coming in contact with the industrial or the pharmaceutical modelers, the in silico modelers. For example, some of the experience we have with AstraZeneca in the diabetes model field, they started to model our chip experiments with a liver pancreas combination, and therefore we started to decrease the number of chips we need in combination with the in silico model to get a real clear result on diabetic appearance in the chips, for example. And the artificial intelligence background also comes from the same level. So our software engineers are now involved, which was not even intended from the very beginning, of automating all the processes into like using artificial intelligence pieces to enrich the data of the in silico in situ model. And that are the 3 big things. Patient, yeah. By the way, patient groups are coming and trying to help us with financing. So because if you not only stick with the medical doctors, which we have obviously in the different projects, but also like extend it to the patient groups, that probably is something we should consider in the next stakeholder meetings and world summits of the next years.
Linda, you wanted to say something?
Yeah, I would say, we use all kinds of machine learning and artificial intelligence on a daily basis, because we use it a lot to conceptualize the biology from the patients, because there's heterogeneous presentations in the patients, and we publish like the first papers that molecularly classify endometriosis, et cetera. And it's super, super important to the work we do to do that upfront conceptualization, because every time we make any kind of the in vitro model more complex, it's more expensive. And so we use that, and we work now with a colleague who does, he's been doing what's called species translation, trying to look at translating from existing multi-omic data sets of mice and people, but now he's helping us with our in vitro models to people, because there's a lot of data sets for humans and animals. And there's ways that you can extract features from data sets where it may not be like an obvious thing. So I'm a strong advocate for including those kinds of what I call systems immunology or systems biology at every single step of the process, not just like, there's a lot of sort of more pedestrian applications of AI, but an informed systems immunology or systems biology at every step really does help you diminish the number of things you have to do, and helps you get a lot more information out of each experiment that you do, because these experiments are so expensive.
Mm-hmm. I'd like to pick on that. So we heard now a lot about the importance of modern data analytics, including AI. And before you brought up, the patient as the source for your tissue. So wouldn't the ideal combination be that you get kind of both? You get the data from the patient, be it patient history or aspects of what has been measured in the clinic plus a sample, and use that to build your chip, which then comes with additional, let's say, sensitivities and aspects to consider about ownership and so on. So how do you see that?
One little thing I'll mention before others chime in, you bring up an important point about ownership. And for us in academia, we can easily form partnerships with clinicians to get the patient samples and to get, do the analyses ourselves and build our own tissue banks that we control. But the consenting process for how we get the samples from the patients often makes it impossible for us to share the models with a company. So we're in a situation right now where we have a really great tissue bank, and then a colleague at Harvard has a great tissue bank on the intestine, for example, and we just can't even bring those tissues to MIT for even just our experimental work that's academic because of the way the consenting was done. And, then we can't share our tissues with a company that we have a collaboration with because of the way the consenting was done. So, certain data we can share, but not any of the tissues. So I think there is a need to think up front from, especially the academics and the clinicians who are building these on how we structure the consenting. There's a lot of need for discussion around easing those data transfers, but especially the cell transfer, because these are very precious resources, these tissue banks.
Other views on that? Otherwise, I would come to a point that was brought up before. Funding is needed. This is clearly expensive research. Now, on the other hand, and that was a point brought up also from the group here, is that also for end users, this is also quite an expensive technology to use. So what are the thoughts around making it more affordable to a wider range of users? What would you have ideas maybe from the engineering side? Yes, Yasuyuki-san.
Yeah, actually, I'm not a specialist of microfluidic, and when I first came into microfluidics, I was so surprised. It's a very laborious work, and now. Still now, I still now keeping that initial impression. So I feel because I just heard that the FDA just widened the definition of MPS, and in that new definition, MPS include almost all advanced cell culture system, including even static. Mm-hmm. So from now on, I think, according to the necessity, for example, context of use, we have to carefully choose the appropriate technology, I think. If it's really necessary, we have to do that, even if it's laborious. But if it's not necessary, we probably just forget about that. So from now on, not only just sticking to fluidics, but also probably we need to re-evaluate the static culture, for example. Mm-hmm. I feel from now on, yeah.
Mm-hmm. Great, thank you. Peter, what do you think? How could your models become cheaper?
I mean, I think there are multiple aspects. In the end, the microfluidic chip is, in most of our cases, not the expensive part. It's the cell part. So if you want to buy standardized iPS cell-derived cell types, those are the more expensive one than the chips. In terms of microfluidic chips with scaling and automation, you can get down the prices there. But here it's also important that and that's especially for us academics, that when you start designing and working on a chip, you have to keep in mind from the beginning that this should be scalable at some point. So you can do whatever you want and whatever your academic kind of freedom gives you, but it's important to think about, okay, how could this be scaled up later on? So I think this will bring down the prices in the scalability. The other thing is, it's often not per se the chip that is the expensive part, but really the infrastructure. And here, I think standardization and interoperability between different systems will bring down prices in the end. And the last point is, it also depends if you look at the cost to what you compare it. If you compare it to a well-played culture of a cell line, yes, then it's extremely expensive. If you compare it to a large animal model, which you would otherwise be using, then it's cheap again. So it really depends on the more you know about a system, the more you know about what you can do with the system and what you compare it to, then it might not be that expensive anymore.
Mm-hmm. Very good point. Thank you very much. Now, switching gears a bit, Alison, we live in the area of personalized medicine and precision medicine. Could MPS play a role there? And if yes, which?
Yeah, no, absolutely. I think, I mean, all the components of the MPS, right? And starting from reprogramming cells from the patient, capturing the entire genome in a pluripotent stem cells and creating the cells models, and then having the robustness of an in vitro system that allows you to detect even subtle variations. Yeah, so I definitely think that's the future. But for us to get there, I mean, there are several things that needs to be done. And I like the discussion about cost, because we need to expand this. To start seeing effects, we need to start looking at different genetic variations. We need to start looking at different genetic backgrounds, working with more patients, more cells. And right now it's just not feasible. So reducing the cost, and eventually, as with any other technology, the cost will come down. But if you can speed it up, that stage where we can grow like 100 organoids from 100 people at the same time, that would be fantastic. Yeah, that's when we got to the personalized medicine. Yeah.
And so when you say it's currently not possible to work with like a large set covering a large genetic background, is that a cost issue, or is it an access issue or consent issue as discussed by Linda, or is it a mix?
Cost. I mean, I have plenty of access. I have so much access, but it's so expensive. I mean, to get and make the tissue bank, and then because you need all the cell types, you need to expand the epithelia, you need to expand the stroma, you need to expand the endothelia, you need to have some way you deal with the immune cells. So we get blood, but we haven't like bit the bullet and started isolating the immune cells from the tissue, so there's just an enormous cost to even processing and creating the tissue bank for an individual patient. And iPS cells offer hope that you could do, make an iPS cell, make the whole patient, but we're just so not there yet for chronic inflammatory disease. I mean, you have to do it for heart, you have to do it for brain, but for the diseases I study, they're not even on the radar screen. So you've got to deal with the primary cells from very well phenotyped patients. And I have access to the patients and the phenotyping. I have more people wanting to give me tissues than I can possibly get because of the cost of processing them.
Wow. So I see we're already coming towards the end of this discussion. So I would like to just ask around as kind of a final question. So given where we are today and what has happened in this rapidly evolving field, how do you think will drug development look like in, say, 10 years? Uwe, do you have an idea there?
Yeah, Uwe, that's yours.
I think definitely in 10 years, the fit-for-purpose assays and the context of use assays are in the routine background for drug development, IND, IMPD. We think that organismoid solutions, this is how we coined or call the human on a chip or the entire body on a chip, developments are all on the organismoid level already there. So we have miniaturized organisms on chips, which are autologous source with all the 11 organs, let's say, and the vasculature from one and the same individual donor, but they are maybe not a disease model yet. So we need then to move these healthy organismoids into disease, but that is our vision. So no doubt that we will have IND, IMPD. We are probably not able to, within that time period, to bring any of these into an ICH guideline, but that is the reason for that is ICH harmonization itself will take another 10 years between the different regulators worldwide. But the individual regulators, especially the FDA and maybe other things, will definitely have voluntary approved or qualified together with sponsors and with the provider industry fit-for-purpose assays in this area. And on the research area, science area, we will be there also. It is expensive now on a miniaturized organismal level. And that will then boost the thing. The costs on all three levels, devices, chips, biological models, and assays bear a significant economy of scale potential and a big economy of scope potential, because the economy of scope with the assays on a disease model is transverting 90% of these chips into in silico solutions. And that is all economy of scope. We need data from these. So I think economy of scale and economy of scope will make it the same process like with human genome sequencing. We started with 1 million, we are now at 1,000.
I'll bet you $100 disease models beat ADME talks to wider impact. I'll bet you $100. So that's my 2 cents.
Okay.
Because the things that you need complex models for ADME talks, you're going to have to develop a complex tissue, and I think the disease. So I'll bet you $100. 10 years. 10 years from now, Adrian, check back with us. 5 years? Was it 5 years? Okay. 5 years.
Let's say 2 years.
3 years. Okay. Well, yeah.
No.
Better start working harder.
December 31.
Okay.
Christmas 2031.
Okay. I think that I see we're 3 minutes to the hour, and we should still save some time for the announcement of the winners. So with that, I think that was a nice ending of our discussion. I would like to thank all of you for your lively contribution. Thanks a lot. And with that, I hand over to the organizers.
Adrian, could you please answer some questions in the chat from the audience to the panelists? And yeah, Ricardo will announce the winner now.
Oh, we still have more time. I thought it ends at 1.
No, just answer, type the answers.
Oh, type them. Okay, yeah, all right.
Thank you.
Let me have a look, sure.
Thank you, Adrian, again, and thank you to all the speakers, the panel, to bring up this interesting discussion on existing models and future technology developments. Now, I do have the pleasure to announce the winner of the Young Investigator. Just give me a second, so I will share my screen. Here we go. I believe you will all see it.
Drum rolls.
Yes, I'll just give a few seconds, as I guess somebody might have some lag. You cannot see it?
No, we're seeing your desktop.
Oh, okay.
There it comes. There it comes.
Okay. Now you see it, correct? Okay, yes. So the winner is Dr. Devin Mayer, as chosen by this audience So Dr. Devin Mayer, congratulations. No, sorry, you're not doctor yet. So thank you for your great talk, and we all hope to see you next year an in-person event. And now, I think in a further citation, I think I will leave the stage to Thomas for the closing remarks.
Okay, I will jump in because I think he had a conflicting meeting and jumped off in last moment. Thank you very much for all 300 participants who joined today and all 570 participants who registered for this event, and I hope they will also watch the recording. So then again, thank you very much, and it was great meeting, and sorry for some technical issues. You still see the Zoom ghost and Zoom travels consistent even 2 years after pandemic. And please follow us with our newsletter. We will be announcing more and more information on our big New Orleans event, which hopefully will be in person in May in New Orleans. We will listen to some nice jazz music there and have great wine and great charts and great talk about microphysiological systems. And we will make sure to move the field forward and make it payable and viable, right? Thank you very much.
Thanks, Lina.
Bye, everyone.
Thanks, guys. Bye-bye.
Thank you. Bye.